Good morning, everyone, for ISGO 2020. This is the second day for our conference. Here in Korea, we have beautiful autumn weather today. So this morning, we will have a session 3, germplasm resources and conservation. And today we have chair, Professor Dong-Lin Jang from University of Georgia. Dong-Lin, here we made the chair badge, so you can start your session. So Professor Dong-Lin, if you are ready, could you please start to talk?
Okay, thank you very much, Kim. And it is a really pleasure to be chair for this section. And I'm from University of Georgia and in US. Now it is about 8 o'clock p.m. here in my time. And I just want to say good morning to the Asia, maybe good evening to America, and good afternoon for Europe. So welcome to the third section of those symposium. And today we have a 1 keynote speaker, 2 invited speaker, and several other speakers. So the first speaker will be Todd West. And Todd is a professor at North Dakota State University and the director of the North Dakota State Woody Plant Improvement Program. He earned his PhD from South Illinois University at Carbondale. The North Dakota State University Woody Plant Improvement Program focuses on the development of new woody cultivars, majorly for USDA Hardy Stone 3 and 4, is cold place. The purpose of the project is to increase breeding efforts and germplasm collections for ornamental woody plants suitable for cold continent climate, because the Dakota is in the middle, which is perfect place for the trying. The program has released more than 60 ornamental woody plants to the nursery industry. And the more important, those releases are doing well and proved to be hardy for their type in the commercial nursery industry. Dr. West is also serves as the president of the International Society of Magnolia. Today, he will share with us ornamental woody plant breeding, selection, and evaluations for cold climate conditions. Let us welcome Dr. Todd West. Todd.
Thank you. Thank you very much for having me. It is a pleasure to be here. I wish I was with you. Unfortunately, with COVID, that's not possible, but I'm there in spirit. So what I'd like to do then is to talk about our program and what we are doing. So the talk I'm going to be giving to today is ornamental woody plant breeding, selection, and evaluation for cold climate conditions. I gave a similar talk back in 2017 in Sweden with part of the Magnolia Society. And the title I had then was Breeding, Selection, and Evaluation for Cold Climate Conditions. A very similar talk. But while I was there, they had several other speakers. One was from Norway, one was from Sweden, one was from Finland, and they were all talking about how horrible it was for them to be able to grow plants. And the more I listened and heard some of the species that they could grow, I was like, no way. I had to change my title, and then I changed my title to My Climate is Worse Than Yours. And that's really what it kind of comes down to, is looking at these climatic differences and how we can work on these woody plants. So here's a picture of me back in 2019, back in January, and it's showing in Fahrenheit. But as Dong-Lin said, is that we're the intercontinental climate, and so it's very cold. We have very strong fluctuations of temperatures, and that can be very harmful to woody plants. So I just want to kind of give you an idea of where I am. So obviously here we are in the US, or the world, and I'm located in the US. So here's our USDA plant hardiness zone map. Go to the center of the US and all the way north to where it is the coldest part of the United States. And that's where you'll find me. When people visit the United States, it's one of the 2 states that people never visit. And I'm one of them. So if we zoom into North Dakota, you can see that it does get quite cold. The northern part of the state gets down to about minus 40 centigrade. I'm in what's considered to be the warm part of North Dakota, right there on the southeast corner in Fargo. So we're in that climate zone of 4A. So we can get down to about minus 34. But in 2018, we got much, much colder. We reached that minus 40 throughout most of the state. And it really harmed a lot of the plants. It was good for me because I was able to really make some good selections. Now for our program, we have several sites that I primarily focus on with selection of materials that we grow out. Fargo is where our main campus is. It's right along the Red River. And then directly to the west, there is what's called Absaraka. And Absaraka is our research arboretum. And this is where we do most of our work. Now we're expanding and we plant in all of the major cities across North Dakota. But I'm really interested in the one that's up north, right by the Canadian border, called Langdon. It's in the northeast corner of the state. And that's always into a true zone 3. So they get down to that minus 40, minus 35, 40. So with our temperatures, we're at that 47 degree north latitude. I use the Köppen-Geiger climate classification to be able to look for plant material that's going to be suitable for us. So we're classified as a DFB, which means it's a warm summer, humid continental climate. Coldest month averaging below 0 degrees Celsius, all months with average temperatures below 22 degrees, and at least 4 months averaging above 10 degrees. No significant precipitation difference between seasons, and that's really what we have. We're very dry, but we can be very warm and humid during the summer. So if you look at that Köppen-Geiger classification across the world, this is what we're looking for. And so North Dakota is that kind of darker blue color. And then I look across the world and see, who is a good match. And so we're looking for, who falls on that 47 degree north latitude. Now, obviously we can go further north, but it's not looking at the elevation as well. I'm very interested, obviously, in high elevation plants as well, and looking for that really cold extreme. This is kind of an overview of what we deal with in North Dakota. North Dakota, we have an average growing days of 130. Our last frost date is May 10th. Our first frost date is September 25th. And here it is, the end of October, and we already have snow on the ground. Average temperatures in 2019, we got down to minus 36 up at Langdon, which is that northern site that I have, almost reached minus 40. But this past winter, we actually were fairly warm. We only got down to minus 30. Langdon was the same. It was fairly warm. It only got down to minus 31. So it wasn't as cold as what we were hoping. But the program has evaluated over 200 plus genera, 3,000 different species and cultivars of trees and shrubs. We've evaluated over 9,500 different accessions starting back in 1974. We also have the largest woody plant ornamental collection in North Dakota and the Northern Great Plains. We have approximately 32 hectares of woody plants that we have. So if you ever use Google Maps and type in Absaraka, North Dakota, this is the little dot that you'll get. If you zoom in, because most of the state is agronomic production, but if you zoom in on Absaraka, just slightly north and east, you'll find this little island. And in this island is my research arboretum. So this is the view as if you were entering the arboretum. We have display gardens. We have all these different germplasm trials from plants that are regional, international, everywhere in between. I'm one of the few people that gets to go visit during the winter months, and it's just absolutely gorgeous. So if you look at the arboretum itself, we have, like I said, about 32 hectares. The center is the main arboretum, and everything is planted out alphabetically by genera. These other areas are the shaded areas. This is where we do our research trials. We do some short-term work, we do some long-term work, but everything's kind of lined out in rows. So approximately about 22 hectares of the 32 are in woody plant at any given time. So this program is not new. It's been around since 1954. My predecessor started the program, or really began in 1971, and he's the one who really established the main part of the program. Introduction started back in 1986, and under his direction, Dr. Dale Herman, he released 51 selections. I started in 2011, and since then, we've actually had 59, but this year is going to be 60. And what I focus on is diversity. And we need adapted, winter-hardy, pest-resistant woody plants suitable in northern climates. Many of the commercial available nursery cultivars are not suitable for this climate because of our cold, but also low moisture and high soil pH. Our pH in North Dakota is right around 8 to 8.2, which is very high. And so we need to increase plant diversity in response to disease and insect pest issues. We've had Dutch elm disease. We now have emerald ash borer. We have a lot of issues with native and introduced species. This is a diagram that shows native woody plant species diversity in the United States. And as it gets darker, so that dark center kind of red color, that is less diversity. And North Dakota is right in the heart of that. So we don't have a lot of native species because we're a central plain state where we have grasslands. So I don't have a lot of diversity to work with when it comes to breeding and selection. So my main two goals is to evaluate unreleased or released cultivars from the nursery trade to determine usability. But then also we increase diversity through selection and breeding. We're very fortunate because many of the selections are suitable for a much wider use. We even have a nursery in New Zealand that is growing our materials as well. We're hoping to try to get more material into Europe as well. But our material is used a lot across the US. Also, urban conditions are very similar to our conditions. Typically, urban soils are compacted, they're dry, they have high pH, and that's exactly what we have. We're a very dry state. We're the 8th driest state in the United States, and our soils, as I already mentioned, are greater than 8.0. So some of the evaluations that we're doing, obviously with germplasm source, we're using foreign and domestic seed sources, growing out populations, seeing how they do. We do plant breeding, traditional breeding, and also using tissue culture, looking at somal clonal variation, embryo rescue, mutagenesis, and a little bit of genetic engineering. We work very closely with a lot of major nurseries here in the US, Bailey, Isley, J. Frank Schmidt, Spring Meadow, JN Plant Selections to trial their material as well. We have a big trial of hybrid maples that are crossed between Acer palmatum and Acer pseudocibolianum, which is one of my favorite plants. Because Isley put out this series called the Jack Frost Collection, which is a hybrid between the two, palmatum and pseudocibolianum. Unfortunately, none of them are hardy. They have ice dragon and Arctic jade and north wind because one of the problems we have in the north is that we cannot grow Japanese maple. And so unfortunately, we don't have all this diverse color and leaf shapes and etc. And so when we trial them, this is typically what I do. My job is to kill plants. And I do a pretty good job at it. Every now and again, we get some nice plants that do survive. We do, as I mentioned, a lot of work with the industry. And so we have a really large block where we test cultivars and see how they do. With plant improvement methods, we do breeding, we do seed source and seed lot variation, and then we do selections by landscape observations. So with traditional breeding, we're working with elm, lilac, magnolia, and maple. So here's me on the right, out there in the springtime, and I am doing some crosses with our maple collection. So I'll just start off talking about some maples. And one of my favorite plants is the Korean maple, Acer pseudocibolianum. We have a selection that is the cultivar Kordak, and it is the hardiest of all of the maples, the Korean maple selections that's available in the United States. This has been proven hardy down to -40. There's quite a few other strains of Korean maple that are sold into the commercial market, and they're marginal. And so this is what we use to fill that void because we cannot grow Acer palmatum. What I like, though, is that it also retains its leaves throughout the winter months, and this helps with winter sun protection. And that's a big issue for us. But it's absolutely gorgeous. You know, beautiful foliage during the summer, and you cannot ask for a better fall color. But again, there's not a lot of variety. You know, with Korean maple, the variation is very minimal. So we did some crosses with Japanese maple and with our Korean maple Kordak, and this is the variation that we're seeing. We're seeing straight green leaf, we're seeing red leaf, almost a purple leaf, cut leaf, non-cut leaf, and a lot of variation. This is one of my favorites that's coming up, and it has a lot of Korean in the background, but it also has that deeper cut leaf, and it emerges out red and stays red for quite a long time, but it has that green center. So this is one we're evaluating for hardiness now, and we'll talk about ways that we evaluate for hardiness. Because we have high pH, we have some soil-induced chlorosis. In this photograph, this is actually an aerial photograph of Fargo, and the trees that are yellow should not be yellow. These are typically our Freeman maples, the cross between silver maple and red maple. And so what we typically see are these yellowing leaves because they're not able to handle our high pH. Silver maple, the Acer saccharinum, shows dieback issues for us. It's not completely hardy. Also cannot handle our pH. Red maple is the same way. We have a lot of iron chlorosis with red maple with our high pHs. So with our evaluation, we've seen there's quite a few cultivars and germplasm that does very well in a high pH soil. And so we're utilizing these to be able to breed together to be able to come up with a more suitable hybrid. So we're using one of our releases called Fall Grandeur. It has a nice upright form, and that's what a lot of city foresters are looking for, to be able to fit into boulevards, into tighter spaces. And we're crossing that with some of the hardy silver maples. So that's what that other picture was. My technician's here on the right, but you can see that we're crossing from silver maple into red maple. We do red maple into silver maple. And then what we do is then we grow the seedlings out. So here's one of our blocks where we've lined the seeds out. And then we walk the rows in the fall and pick the ones that have the best fall color. Once we make those selections, we pull those individuals out. We plant them in our arboretum for long-term evaluation. Here's one of my grad students on the right. She's evaluating the colors. Now, magnolia has a special place in my heart. I love magnolias. As Dongling said, is that I am also now the current president of the Magnolia Society International. So I do a lot with magnolias. And we do quite a bit of hybridizing with them. So obviously here's me again, pollinating magnolia flower. Because my predecessor made a selection of Spring Welcome magnolia. It's a cross, it's a lobneri type. So it's a cross between stellata and cobus, and has done extremely well for us without any problems with pH, moisture issues. and with hardiness. And most importantly is that it's flower bud hardy all the way down to minus 40 degrees centigrade. And so we don't get bud kill. Now, the issue though we have is that we can grow a really nice white magnolia, but that's it. So one of my breeding objectives then is to increase color, trying to bring some yellows, the pinks, the magentas, especially the yellows with the acuminatas, because they bloom so late, so we can avoid late spring frosts as well. So that's what we're looking at, is that bloom time, late and extended. The top right picture is a magnolia hybrid that Dennis Ledvina from the United States released called Rose Marie. And it blooms fairly late, and it has a very extended bloom time, and it has that beautiful magenta color. And this is one that we're using as a parent then to cross in with our spring welcome to be able to get, hopefully, a nice, hardy, colorful, late blooming magnolia. I'm also very interested in uprights. I like getting more compact plants as well. And one thing I learned from breeding magnolias with Dennis is that you want to make the cross from the pollen parent with the color into the other magnolias. So we always cross color into our spring welcome to make sure it's there. If we do spring welcome across, we won't really get the color that we're looking for. So we collect pollen. Breeding magnolia is one of the easiest plants to hybridize. So we collect pollen. We take the anthers off, place them on paper, and allow them to dehisce, open up. They'll release the pollen overnight. So this is typically what my hotel room looks like. I always have paper with me and envelopes to be able to store the pollen. I'm always worried what the cleaning staff will think. Here in the US, we have a TV show called Breaking Bad where they make drugs. And I always figure that the hotel staff probably thinks that I'm making drugs, but I'm just collecting pollen, something very safe. So we do a lot of field breeding. On the right is my son. He's actually one of the best hybridizers I've ever worked with. He gets such a great seed set. It's just amazing. So I always take him along on my expeditions. But one of the problems we have in the field is timing. Weather is unpredictable. Bloom could be happening. When I say now in North Dakota, means, it's generally happening, and then we'll have winter. We'll have late snow, and it just is not very conducive. And with cold temps happening in the spring at night, that night cold temperature causes the pollen tube development not to really advance. And so then we end up with poor seed set. So it's very unreliable. So we've switched over now to forcing flowers in the greenhouse. So I have an extensive collection of potted magnolias. And this allows me to control the timing of the flowers. It also helps me to have increased temperatures for consistent pollen tube development. We also have limited pollinators. So obviously I do all the pollinating, so I know exactly what's going on. Also, I don't have to climb a ladder. I'm getting older and I don't want to break my leg. The big disadvantage though is the space for the number of plants, and having numerous cultivars as well. So that's the big disadvantage. I can't have a big collection to cross into. Once we make the crosses, fruit will set, seed will set, we'll collect the seed, we'll clean the seed, we'll stratify the seed, and then we grow it out. So we have thousands and thousands of these little plugs that we grow, and they're all these different crosses. And we let them grow out for about 1 to 2 years, and then we put them out in the arboretum and let nature take its course on whether it's going to be hardy or not. And then we have to wait for flowering. With seed source and seed lot variation, we get a lot of seed from all over the world. Obviously, it's more difficult now as borders are closed and closing for germplasm. Years ago, especially with my predecessor, it was much easier to get material from China, from Europe, and now it's very difficult, sometimes even impossible. But here we have a native swamp white oak, Quercus bicolor, and typically does not have very good color. And the one we are seeing, it's getting a really nice red fall color, and it's pH tolerant, whereas the other ones are not. Another native species for us is Cercis canadensis, redbud. And again, not very hardy, but we have a seed selection that we have that is doing very well. And it's been released through J. Frank Schmidt. And they say it's hardy to zone 3B, getting into that minus 35, but it's really only hardy to minus 4A. So we're getting into that minus 30 point, but blooms quite consistently. Then with selection by landscape observation, again, we just drive around and let the communities, because the city foresters here in the US, they plant a lot of seedlings along the boulevards and allows us to make selections from that genetic variation. So here we have in the center here an upright Baroque, which is very atypical for the species. So we select that, we'll select seed from it, we'll select cyanwood, we'll graft them, trial them, then work with commercial nurseries. Here's one of our long-term evaluations that we've been looking at. This was selected and released back in 2015. We call it September Flare because for us, this sugar maple colors every year in the middle of September. And if you look in the background of these pictures, there's nothing else that's really coloring for fall color except for this. So it has an early shutdown, and I look for early shutdown in plants because that's a key indicator that they'll be most likely hardy. Now, one question I get a lot is how long does it take to release a tree? And for us, it takes a long time, upwards to 20 to 30 years before we actually will release, and it generally has become available. I borrowed this slide from Chris Bachtel from the Morton Arboretum, which is just outside of Chicago, Illinois. And they have the Chicagoland Grows program. And he put together this presentation, which I really like, because it gives you an idea of how long it takes. And so if you look at the oval on the top left there, it says the time frame 15 to 50 years. With the research and development and then the time it takes for nursery production to happen, it can take anywhere from 15 to 50 years for a woody plant, especially a tree, to make it into the landscape. And that's a long time. So it's very dedicated research. That's why it's exciting to see where we're starting to get molecular markers. You know, we're starting to get these extra tools now to be able to shorten and direct our research efforts. So some recent selections that we had out of our program. One that I'm very excited about is Northern Empress Japanese elm, the Ulmus davidiana variety japonica, cultivars Burgundy Glow. It's a smaller elm than typical for the species, but has really high quality foliage during the summer. Now for us, Dutch elm disease is a really big issue. And the Asiatic elms are resistant. And so we really look towards those Asiatic elms now. But the issue with elms is that we typically don't see very good fall color. And everybody wants to have fall color. So with the Northern Empress, it was selected because it has this apricot to burgundy red fall color. And it's just absolutely gorgeous. Very reliable every year. Then one of my very, very favorite plants is this Betula costata, which we refer to as a dwarf Korean birch or just Korean birch. But this is a dwarf selection. It's called Cinnamon Curls. And it has, again, beautiful foliage during the summer months. This tree on the left is the same as the right. The right is the fall color, so you get a beautiful golden fall color. Never been pruned. This is how it naturally grows. And then it has amazing peeling, exfoliating bark during the winter months. So you get this really nice season extension, beautiful foliage in the summer, outstanding fall color, and then beautiful bark and beautiful form. A little background on this plant. We published the release in 2016. It's published in Hort Science. So if you want to read about it, you can. But it was selected from a population of seedlings obtained from Longwood Gardens out at Kennett Square, Pennsylvania. And Longwood Gardens sponsored a collection trip with the National Arboretum to collect this material. And there was originally 34 seedlings from this lot that were propagated from this original collection germplasm. And this was done back in 1985 when we got the material. And one seedling out of that 34 had this unique habit of being a dwarf habit. So further with the background, the original material with the National Arboretum here in the United States was sponsored with Longwood Gardens to collect, and it was collected back in 1966. And it was an expedition done by Corbett and Leidy, who worked with the USDA out of Beltsville, Maryland. And they traveled to South Korea, the Republic of Korea, and collected seed. And please forgive me, but they collected on Mount Deogasan. My Korean obviously is horrible, and I do apologize. Please don't hold that against me. But they collected the seed, brought it back to the United States and grew it out. We got seed, a second generation then, and grew it out. And we found that we had this one that's dwarf. We have other selections from that same collection. You can see where I'm standing by it on the left. This is what the species should look like. But on the right is what this one seedling is doing, and it's staying very compact. And we've been working with Isley Nursery, and they've been doing cuttings of this now, and they have it available in the nursery trade. And this is just a close-up, and it's already showing, even after this short time, this nice exfoliating bark. Another release that we have is called Lava Burst. This is a seedling selection that we found, and it's a upright of Aesculus glabra, which is atypical then of the species. The species is generally as wide as it is tall, but it has very short internodes, and the foliage shows greater resistance to leaf scorch as well. So the summer months, it gets an absolutely beautiful fall color. It has that 5 leaflet palmate leaf, and it produces very little seed, which is good and bad. You know, good for the landscape and worries about invasiveness, but bad for us to do any more breeding work with. But planted out, it has this nice, tighter, upright type growth. Here it is on the left as a first year planting stock. On the right is after 2 years. And then this is what the fall color looked like in the form this fall, just a few weeks ago. Another one that we look at and I'm very excited about, because we are always looking at disease pressure and insect pressure. And we have bronze birch borer, a native borer, very similar to emerald ash borer. It's a cousin of it. And it really devastates our introduced birch species. And this is a newer one for us, is Betula tianchanica. This is an Asian species and absolutely fabulous. I love it. And we selected this one and called it Emerald Flare. It has beautiful bark and has a little bit of exfoliation to it. We introduced it into tissue culture, and we have a grower that's growing it out now. And just absolutely beautiful. What they're finding is that with birch, typically tissue culture liners don't grow first year. They're just more, they do establishment. And then the second year, they put on all their growth as a tissue culture liner. But they're finding that this emerald flare starts growing in the first year. So it cuts a year off of production, which is really exciting. Azaleas is one that typically does not grow in a high pH soil. We had quite a bit of germplasm collection of azaleas, and we found this one is able to handle our high pH and extremely hardy all the way down to zone 3B, so it's able to handle minus 35. And we're working with a grower to tissue culture and to propagate this to be able to get it out into the trade. One species that is becoming very popular in the United States is Syringa reticulata. And that's because it has very few disease problems. And here in the US, as soon as something shows very few disease problems, it starts becoming very common in the landscape. People produce it and use it quite extensively. And so we've made a release of this summer flare Japanese tree lilac, and this is an upright tree lilac. Ivory silk is the most common that's planted. It's about 6 meters tall, whereas summer flare is about 10.5 meters. So it's quite large in comparison, and it's a better selection for when you want to have a very large tree, because typically Japanese tree lilac for us, they're not a very big tree. And so we have quite a few evaluations going of this. We have it grafted. We've been putting it onto boulevards to see how it does. And it usually grows at least a meter per year. It's quite a vigorous grower. We have quite a few selections of evergreens as well. And one of the common evergreens that are in our landscape that's really hardy is mugo pine, Pinus mugo. And one of the issues, though, is that mugo pine is fairly slow growing. It's one of the slower of the pines. So we made this selection because it actually grows faster than the most common cultivar that's in the trade called Tannenbaum. It also has a darker green foliage color. So our Highland Splendor is on the left and Tannenbaum is on the right. And we found that it grows 20% faster than the Tannenbaum cultivar. So the nursery trade is very excited about that faster growth rate, because then they can produce a bigger plant, which then demands a higher value. Some other past selections, we have a Northern Flare sugar maple. Again, sugar maple for us is not hardy, borderline hardy, but then also the big issue is the pH. And this one is pH tolerant. It's also drought tolerant. And so it has a really high quality foliage during the growing season, and it has a really nice fall color as well. And it shuts down early, and that's where we get that hardiness component from. We also have an Acer triflorum because we cannot grow paperbark maple, the Acer griseum. So this is our substitute, is the 3-flower maple. And we have what's called orange aglow, because there's the color range for Acer triflorum ranges from red to orange. And our selection that we made is this orange. And so it is a really nice selection that has that beautiful exfoliating bark and outstanding fall color. And this is our replacement for Acer griseum. As I mentioned earlier, birch is a very important genera for us. And so we have one that's Prairie Dream. This is a paper birch native to the US, highly resistant to bronze birch borer, this selection, where normally the species is not. And it also has this amazing, outstanding, very bright white bark. We also have this Dakota Pinnacle Asian white birch. It's the platyphylla, and a nice narrow upright selection. This is very popular in the US and in Canada. And then another native, Betula nigra, which again is not hardy, but also has severe chlorosis issues. And we made a seedling selection from a germplasm source that does not show any chlorosis for us. Now, another one we've selected, now this is germplasm that we acquired from Harbin, China. And Harbin, China lies right on that 47-degree north parallel. And so the climate is very similar to our climate. And so the Prairie Horizon, the cultivars Harbin, does extremely well in these continental climates where we have humid, hot summers, but extremely cold winters. And we're seeing a lot used here in the US in these northern climates. We also have a Eucerian pear. The calorie pears are not hardy for us. So we've made selections using the Pyrus euceriensis, and they're much more adaptable to the high pH and with our colder climate. We're also doing some oak hybridization. This is an oak, a white oak crossed with a English oak, because white oak is not able to handle our high pH, whereas Quercus robur can. And this hybrid does extremely well. It still maintains the Quercus alba color, but it has the pH tolerance of the Robur. And this is one that's doing very well in the trade. Honey locust is a big one for us, native to the US, and it's extremely urban tolerant. This is one of the top trees used in urban settings. And this is one of the hardiest. And I've actually been told by the nursery trade that not only is it hardy, but it's also one of the fastest growing in the nursery. And so they really like it for that aspect. I had mentioned elms earlier with our Northern Empress Japanese elm, but we also have an American elm that is showing to be tolerant of Dutch elm disease. And so we're starting to get some of these DED resistant cultivars. And this one's extremely hardy and doing extremely well. Here you can see in this picture on a boulevard in Fargo how deep green it is. In the forefront, the tree on the right, that's a Celtis occidentalis, a hackberry. And so you can see the color differentiation between the two and how deep green an American elm is. A couple more evergreens, Royal Splendor Norway spruce. This is actually most likely a hybrid, and I need to do a genetic evaluation on this because the seed was collected in Finland, and it's where Siberian spruce and the Norway spruce meet, where their two native ranges overlap. And so they've referred to that as a Finnish spruce, and I think this is actually a Finnish spruce. And thinking that with that Siberian coming in, this is completely resistant to all needle cast disease. And that's the fungus that really plagues us here in the United States with spruce. And so this one in the nursery trade has got people really excited. It has beautiful foliage, shorter needles than what's typical of Norway spruce, and just absolutely fabulous. Pinus sembra is one of my favorite pines. It's one of the slowest growing pines as well. It was selected in 2015 by the American Conifer Society as one of their collector conifers of the year. But Pinus sembra has that beautiful upright growth. And as I mentioned earlier, I love more of those upright type plants. And so here is a young planting of it, and all the growth just continues upright. For us here in the United States, there's a big push towards upright plants. This is one of our natives, Platanus occidentalis. Again, not hardy, but we selected from its native range, from the most northern part of the native range, and selected them, and we came up with this. And beautiful exfoliating bark. Some potential future selections that we're making is a Japanese Katsura, Cercidophyllum japonicum. One that I'm really excited about are these truncatum hybrids. J. Frank Schmidt released this Sunset series between truncatum, the purple blow maple, and then the Norway maple, but they're not hardy for us. But we have a source of Norway maple that came from Finland. And we also had a Acer truncatum that was located near this plot. It blew over in a storm, so we collected as much seed as we could off from it to save the genetics. And then we found that it most likely then crossed with the Norway, because we're getting all the variation, but we're also finding that they're extremely hardy. So this was a couple falls ago. They've been hardy down to -36 so far, and we're seeing all this variation. This is one that we're seeing that I'm extremely excited about, is this hybrid. The leaf is much smaller, and the picture on the left is what you typically see with this hybrid leaf. And the leaf on the right is much smaller and it's crinkled. But what's exciting is that the new growth comes out this bright red in the springtime. So all that flush of growth on this tree is going to be this bright red. It does fade into a green, and then it gets this kind of orange-red fall color. We've also made a selection of this purple. All the new growth is purple. It does fade into a green in direct sunlight, but then it will turn a nice kind of bronzy color in the fall. We also have this upright mugo, which is kind of atypical. It's either Pinus mugo subspecies uncinata or Pinus uncinata, depending on what taxonomic condition you consider it to be in. But a very nice tight upright. And again, the nursery trades here in the US are really excited about this one. Now, with some research that we're doing, we do a lot of tissue culture work for propagation purposes, but also mutagenic breeding, such as chromosome doubling. Also, we use the EMS to do some knockouts as well. So here I've got magnolia. We do a lot of shoot forcing. So I'm just getting the vegetative buds to force out. As you can see here, now they have forced out. We then select the shoot. We'll introduce it into culture. And then this gives us our population to be able to work with for mutagenic or just propagation. We do a lot of grafting studies. We're looking at tolerance using different types of understock. And we use our native species, the Quercus macrocarpa, the bur oak, is super tolerant of high pH soils. So we put oak that typically is not pH tolerant onto macrocarpa, and we find that it does extremely well. We do a lot of cutting propagation as well. We know that Acer triflorum is very difficult to clonally propagate. So we've been working on different methods to increase rooting, including foliar applications of PGRs. We use a lot of different treatments to be able to increase the rooting so that way we can get a viable propagule. We do a lot of freeze testing. The outdoors is not very reliable, so we never know how cold we're going to get. So we test a lot of the tissues using programmable freezers. And so here's my grad students making measurements. They're measuring electrical conductivity after potential cell bursts as a result of cold temperature issues. So here we were looking at magnolias. We were comparing saucer magnolia with our spring welcome. And what we found is that the buds are extremely hardy compared to the saucer magnolia, which we were expecting. We also found that the flower buds were extremely hardy as well, and also the stem. So everything about our selection was much hardier than the saucer magnolia. And so that's what we do. We do a lot of these pre-tests to be able to tell if our germplasm is going to be able to handle our harsh winters. So we can really screen the material. And the same with then the genetic manipulation is that, we're not doing any genetic modification, but we are doing kind of these knockouts. And that's why we're excited about getting some of these genomes sequenced so we can start developing molecular markers as well and couple them with these freeze tests. So that way we can really start making better selections, more precise selections, and then potentially cut down on that production time. So I'd like to thank you all for this time. I really do wish I was there joining you. I was looking forward to that, meeting all of you and interacting with you and really getting to see all the amazing things that you have to offer there. I hope in the future that I'll be able to come and visit. The Republic of Korea is on the top of my list. I've been dreaming to go to South Korea for many years. If you want more information about my program, or if you're just interested in interacting with me, my email's there at the bottom. But any of this information is also available on our website. So if you want to know more about our introductions, want to know more about our program in general, those links are there. So I'd like to thank you all for this time and for this opportunity to present.
Thank you very much, Dr. West. It's impressive. I never think about in the cold, you produce so many plants. It's amazing. It's amazing.
It is amazing.
So I just look at my plants, no one can survive in your place. It's just amazing. Yeah. Any questions from floor? Any questions? Because I cannot say the floor. Any question from online?
Yes. Professor West?
Yes.
Yes. I saw a potted magnolia with flowers. How do you get that dwarfed magnolia?
Well, what we're hoping is that we can continue to propagate them and get those colors to show. Now, we haven't, all of the seedlings that I have growing, they're almost ready to mature. So we haven't been able to see the color. So we're not sure what we're actually going to have yet. Because unfortunately, like with our spring welcome selection, it took 13 years for it to bloom.
So you mean it's plant seedling? The dwarfed potted magnolia?
Yeah, potted magnolia.
Yes, potted magnolia.
Well, just think about how do you force the flower?
So how do I force it?
Not force. How do you get a dwarfed magnolia? The dwarfed.
Drop.
It's a small tree with flowers.
Yeah, so what I do with the potted magnolias, I keep pruning them back.
Pruning back.
Yes, to keep them into that potted size. But one thing we're doing is that there is a cultivar called Genie, and that actually is a dwarf magnolia, and we're using that as well to try to get the dwarfing gene into our population.
Oh, I see. A higher cross.
Yes.
Cross the dwarfed.
Yes. And that's where I'm excited, where magnolia is starting to finally get some of the genomics done. So now we can start doing some looking for those molecular markers, such as like with Genie, can we identify that dwarf gene?
Okay. Thank you. Thank you very much.
Yeah, no, thank you.
Any more questions from online or floor?
Yeah, I'd like to ask to Todd.
Uh-huh.
Yeah. Todd, it's very nice to show us your presentation. I'm more interested in the Acer palmatum, which is habitat mostly in Korea and Japan. Do you have any some collection system of germplasm from Korea or China, Japan, into your laboratory?
No, and unfortunately, no, we do not have really a good source of germplasm from Korea, Japan, or China. That's one thing I would love to be able to try to get some of that germplasm in, because what we found is like with our straight Pseudocibolianum, I mean, it's extremely hardy. And I think if we could find some other materials, we're very limited to the actual germplasm that's here in the US, with limited really to the cultivars that have been released. So I would love to be able to see about how we could get some more germplasm into our program.
In the future, about the germplasm exchange, about your material and our material as well. But you have treated some EMS chemical mutagenesis on ESA as well?
Yeah, we're using the EMS. Mainly what we're trying to do is get more the knockouts to get dwarfing, especially in the shrubs. And that's a lot of what we're using the EMS for.
Progress of the EMS mutagenesis treatment on the mutation breeding into the new cultivar already made, or just in progress?
Could you repeat that?
Yeah. How about the progress of your mutation breeding using chemical treatment into new breeding new cultivars? Is it already made it from your laboratory?
Yeah, so we're definitely using that EMS to also try to get a variation within the germplasm as well. And so we are using that for the knockouts, trying to make some selections, but then also trying to breed back. One thing we're doing a lot too is working with the doubling of chromosomes to try to get tetraploids, because the big thing here in the US, in a lot of places obviously, is the invasiveness and sterility. And so we're utilizing that as well to try to come up with abilities to be able to back cross and make some sterile hybrids as well.
Thank you very much. Yeah.
Oh, thank you.
Any more questions? Well, if no more questions, thank you very much, Dr. West. It's
Oh,
very
thank you.
impressive for the work. Thank you.
Yeah, and if anybody's ever visiting in the United States, stop by and see me. Preferably in the summer.
That's great. Well, we continue the program. We have 5 minutes already. This is good. I think it's better than that. And our next speaker will share with us about Japanese Azalea germplasm, the living horticulture properties. And this will be presented by Professor Nobel Kabayashi. This is invited speaker. Professor Nobel Kabayashi received his PhD from University of Tsukuba in 1997 with study on evergreen azaleas. This is a lot of azaleas. For the next 3 years, he worked as curator of Taguchi Gaoka Park. I remember to have train station there, Taguchi. The traditional azalea garden in Gama, Japan, was focused on azalea breeding and the maintenance of heirloom azalea, which is more traditional azalea. From 2000 to 2003, he was selected as a Japanese International Cooperation Agency expert and in charge of the horticulture development project in Argentina Republic under the International Trademark Association. He invested how to utilize the wide germplasm resource of ornamental plants for breeding, which is everybody, we wanted to do that. He returned to Japan and landed as a assistant professor in Kyoto University in 2003. Kyoto University is amazing. I visited a couple of times. It's very impressive. Now he serves as a full professor in Faculty of Life and Environmental Science at Shimane University, with emphasis on plant breeding and the genetic resources. He's currently working on evaluation of plant genetic resource as a breeding material, use DNA markers, and the phenotype characteristics. He also worked on the new cultivar breeding, use local genetic resource, and application for regional revitalization. He tried to grow in more azalea. Let's welcome Dr. Kabayashi and for share with us about azalea germplasm. Dr. Kabayashi.
Hello everyone, this is Nobuo Kobayashi, Shimane University, Japan. I have talk of Japanese azalea. The title is Japanese azalea germplasm, the living horticulture properties. Firstly, I express my respect for the ISGO organizers to hold this symposium under severe situation. And also thanks to have a good chance to introduce you the Japanese azalea through my research. As you know, evergreen azalea is Japan origin ornamental plants. This is the oldest azalea garden, the name Tsutsujiga-oka Park, 6 kilometers north from Tokyo. The old trees of historical azaleas are maintained. 20 years ago, I belonged to this garden as a curator. This is a picture of the enjoying the azalea blossom in this garden. Not only sakura cherry blossom, azalea is also Japanese spring tradition. This is a traditional flower exposition in Ghent, Belgium. The beautiful flower is old azalea, and they are originated from Japan. The natural habitat of Rhododendron ripense, Japanese name Kishitsuji, the meaning is riverside azalea. Besides this azalea, we have a lot of wild azaleas based on development of cultivars. Now I show several Japanese wild azaleas. Again, this is rhododendron ripense with pinkish purple flowers on the riverside rock and the natural flower variations. Rhododendron kenferi with red flowers. The Japanese name Yamatsuji. The meaning is mountain azalea. Kenferi have the wide distribution area and the most common wild azalea. This is macrosepalm, pink larger flowered. The Japanese name Mochitsuji means sticky azalea because of the glandular hair of sepal and young leaves. Rhododendron Cusianum with small pinkish purple flowers, endemic in high mountain of Kyushu Island, and the natural flower variations with white flower mutant. This is Rhododendron Scabrum with large red flowers, endemic in Ryukyu Island. This is a distribution map of evergreen azalea in Japan. So-called evergreen azalea is a species belong to genus Rhododendron, subgenus Tsuji, section Tsuji. There are 17 wild species of evergreen azalea in Japan. They are contributed to develop evergreen azalea cultivars from old period. Based on these wild azaleas, several cultivar groups, Edo Kirishima, Kurume, Satsuki, Hirado, Ryukyu, and Belgian pot azaleas were developed. This is a famous Korean literature, Yanfa Sorok, edited in the 15th century. In this historical document, there is a description of beautiful azalea bonsai sent to the King of Korea from Japan. This is one of the evidence of cultivars developed in this period in Japan. During the Edo period of Japan, when the domestic peace was maintained, Japanese culture, including horticulture, bloomed. Azalea cultivation boom had been raised during the late 17th century to the early 18th century. This is the ukiyo-e art of the garden shop in Edo, now Tokyo. This is the first monograph of azalea in the world, edited in 1692 in the time of azalea boom, 300 years more before. The book titled Kinshu Makura, English translated as a brocade pillow, including 337 cultivars with illustration. The meaning of this pillow book is we can see gorgeous and beautiful flower dream if it put on bedside. Good name. This is the first page of this pillow book. The most popular azalea at that time, Kirishima, is cited. This Kirishima is corresponding to the present Honkirishima cultivar. Now we call this Kirishima related group as Edo Kirishima cultivar groups. This group includes all the cultivar of small flowered azalea. I introduce other cultivar groups. The famous Kurume azalea were developed in the end of Edo period, based on Edo Kirishima cultivars. Hirado azalea was developed in the old trading city of Hirado in Kyushu. This large flowered azalea was based on rhododendron scammler from Ryukyu Island. Ryukyu azalea was developed based on rhododendron ripense and rhododendron macrosepalum. This large flowered azalea is one of the oldest garden azalea. Omurasaki and its sports cultivars. This large purple azalea is most important garden azalea in Japan because they have environmental tolerance and gorgeous flowers. In rhododendron macrosepalum, the mutants of flower shape and color were collected from Edo period and consisted the cultivar group. In rhododendron ripense, the mutants of flower shape and color were collected and consisted the cultivar group. Satsuki is famous as bonsai materials with flower color variations. This group was developed based on rhododendron indicum and rhododendron eriocarpum from Edo period. Oyama tsutsuji is one of the important azalea in old gardens. Recent study revealed this was the hybrid origin between rhododendron kenferi and Ryukyu azalea. The varietal Japanese cultivars have been introduced to Western countries as ornamental new plants and used as breeding materials. This gorgeous pot azalea, so-called Belgian pot azalea, was developed in the 19th century based on Japanese azalea and rhododendron simsi from China. I hope you can understand Japanese azalea germplasm. In the application of these azalea resources, we are conducting these research subjects. I will introduce pick-up topics. The origin of cultivar Kirishima is these wild azalea populations of Kirishima Mountains in Kyushu. I investigated the origin of Kirishima azalea in this field. In these mountains, rhododendron kyusianum, pink-purple small flower distributed in higher area, and rhododendron kenferi, larger orange-red flower in lower area. And the natural hybrid populations were in the intermediate elevation of the mountains. In phenotype, you can see wide flower color variation in hybrid population. And these varietal resources were used as the basement of old cultivars. The chloroplast DNA marker can divide two species. The hybrid population are consisted by mixed patterns. Here is. This is one evidence of hybrid population. Next topic is the genetic diversity and environmental factors of this riverside azalea, rhododendron ripense. This wild azalea distributed in riverside of West Japan, and it have some interesting trait. When the river flood, all the plants down in the water some days. They have also flower color variation. We researched genetic diversity of rhododendron ripense based on morphology and DNA variation. Depending on the distribution area, leaf shape, is different and local genetic diversity was detected in chloroplast DNA. In general, azalea is acid plant. The soil should be acid condition, and if not, azalea shows strong chlorosis by iron deficiency. I investigate the soil pH of root zone in each azalea habitat. Compared to other azaleas, the highest value pH 5 to 7.8 was detected in Rhododendron ripense. It can say lime tolerance wild azalea. Root development were evaluated. Omurasaki and Shiroryukyu vigorous landscaping cultivar developed vigorous root system in both soil conditions. Here it is. And the ancestral parents of these cultivars are Rhododendron ripense. The local species Rhododendron ripense is an attractive wild azalea as also for breeding resource, lime tolerance, cold hardiness, water and drought resistance, autumn flowering. A new breeding program using Rhododendron ripense are running in my field. And in our recent research clarified Japanese large flower cultivars and Western pot azalea cultivars have chloroplast DNA of Rhododendron ripense. The ornamental value and environmental resistant trait originated from Rhododendron ripense may be contributed to the cultivars. The wild species of evergreen azalea were divided into red flowers having only cyanidin series pigment and purple flower having both cyanidin and delphinidin series pigment. The cultivars developed by interspecific hybridization increase their flower color variation based on varietal pigment composition. The wild azaleas and cultivars were able to classify red to purple according to color chart. The key role genes of flower color, flavonoid biosynthesis genes, were isolated, and gene expression and anthocyanin pigment composition are researched. As next topic, I'll introduce flower form mutation and its application. The study of using accumulated flower form mutation in old cultivars like these flowers. As you know, the identity of floral organ has been explained by ABC model. The MADS gene box gene information corresponding to flower form mutation are obtained. The forcing force, and we focused on the long-lasting flower trait called misome show. This flower color lasting more than 2 months with color change. This trait has been found in several species, Rhododendron kenferi, indicum, macrocephalum, also in pillow book. In the expression analysis of MADS box genes, class B AP3 gene is not expressed in the long-lasting flower. It can say long-lasting corolla is caused by the sepaloid petal by homeotic gene mutation. In the cross test for breeding, the long-lasting cultivars have high fertility and can use as breeding parents. From the phenotype of progenies, the long-lasting trait is a recessive inheritance trait. This is a new cultivar candidate selected from long-lasting hybrid. This is Kenferry type. And they are the other candidate of macrocephalum type. You can see the temporal color change. Next, this narrow and spirit flower, the spider type called Saizaki, exists in Japanese cultivars, also in pillow book. We research the morphology and the gene mutation. The new cultivar King Kusak is released from Hortino brand in European flower market. Japanese spider type were used as cross parents. The morphology of macrocephalum and its Saizaki cultivar was investigated. Compared to the normal flower, all Saizaki cultivars form the 5 narrow independent petals, but the leaf shape is different depending on the cultivars. We found out the spider flower is recessive inheritance trait. In the backcross of spider type, the progenies of normal leaf and narrow leaf were segregated to 1:1. Finally, I talk about the conservation and application of Azalea resources. From 15 years ago, we started the research of old Edo Kirishima trees in private garden in Noto Peninsula, Ishikawa. Here is. This is one of the private gardens of Azalea. More than 500 old tree specimens were discovered in this area. Such a large number of old Edo Kirishima plant distribution is unknown in other parts of Japan. According to the old literature, Kirishima existed in this area in 18th century. This is the biggest Kirishima tree in the area, estimated 300 years old. The old azalea in the private gardens are open in open garden event in flowering season. The undiscovered old azaleas are now used as important touristic resources in this area. This is the old azalea. She has a movable own house for protection from snow damage. This is also another Kirishima tree. The local people are conducting the project of preservation as living cultural heritage and utilization as touristic resources. Now I could introduce you the picked up essence of my azalea research. The endemic azalea species and the traditional cultivars are the most worthwhile genetic resources of Japan. In the traditional cultivars, the mutational genes of flower color and form have been accumulated. They are the living horticulture properties. It is needed to maintain the azalea germplasm up to the next generation as the cultural heritage and as the research materials of horticulture science. Detailed reference you can see in the ACTA book. Acknowledgement to all my colleagues. 小ますみだ。Thank you.
Hello. Well, is there any questions for Dr. Kobayashi? This is amazing azalea. I love to say those diversity and the sense is amazing. Cannot wait to say those azaleas. Any question from floor? How about it online? Anybody? Well, Dr. Kobayashi, I have a question. When you, when you study flower gene, what is the major gene you focus on?
Sorry.
When you study the flower gene to control the flower,
Flowering gene, yes.
and the Medax is kind of a family. Do you focus on any specific gene? Any special gene?
Flowering gene concerning about.
Yeah.
Of course, the FT also major flowering gene isolated from
Okay.
azalea. Also same as other plants.
Okay. Do you think about edit this gene, like gene editing? For the flower, yeah.
Yes.
Okay. Any other question from the floor? Thank you.
Yes.
Go ahead.
Professor Kobayashi, I have a question about the flower form.
Yes. Please.
Yeah. Horse in horse and double flower.
Yes.
What are your question? Horse and horse?
Yes, is the same gene of AP3 or different mailbox gene?
Yes, the horse in horse is mutation of PI gene.
PI
PI
gene.
gene. Okay.
How about
class B?
PI
Class
gene is
B.
a B function.
Yeah, B function. In case
AP3?
and in case of the double flower, the AG gene in C
gene.
function, C class.
Yes. How about the AP3, the B function?
Yeah.
You did the expression of AP3.
AP3 is class B.
Yes. What's the function?
Yeah.
What's the phenotype of AP3?
AP3, the stop of AP3 gene expression is the main. The stop of the AP3 gene cause long-lasting flower.
Okay.
Long-lasting flower. That is sepaloid petal. The petal change to sepal. Green petal.
Green petal. Yeah. Okay.
Green petal.
Thank you very much. Thank you.
Thank you.
Any more questions from the floor or online? Well, thank you so much, Dr. Kobayashi. This is impressive.
Thank you so much. Thank you. Good occasion. Thank you.
Yeah. Thank you. Well, before you get a coffee break, we have one more speaker, and who will talk about the dye tracking and the physical dormancy break in seed of Cornuteria paniculata native to Korea. It's authored by Seung-Young Lee, Shi-An Kim, and Kang-Ho Chong. The presentation will be presented by Seung-Young Lee from Andong National University. Dr. Lee, it's yours.
Okay, hello, my name is Seung-Young Lee. I'm assistant professor in Division of Horticulture and Medicinal Plant in Andong National University in Korea. My presentation topic is dye tracking and physical dormancy break in seeds of Cornuteria, native to Korea. Cornuteria paniculata is deciduous arborescent belonging to Sapindaceae family, and it grows only in China, Korea, and Japan, and thus the endemic species in East Asia. It has a long flowering period from June to August and has environmental tolerance such as drought and cold. And it has been known as honey plant because honeybees and short-tongued bumblebees heavily work for nectar. So this is a very good ornamental plant for pollinator garden. So we conducted this research for 3 questions. First one is, do the seeds have dormancy? And second one is, if so, what is the requirement for dormancy break? Actually, previous researches indicated that the seeds had dormancy, but we determined the type of seed dormancy in detail in this research. So firstly, we incubated the seeds under 4 temperature regimes for 4 weeks, but none of the seeds germinated within 4 weeks, as previous researches had indicated. Because harvest time is one of the important factors for dormancy induction or break, we incubated the seeds with full maturation timing, but none of the seeds germinated regardless of harvest time, indicating deep dormancy, as you can see in the pictures. This is a classification system for seed dormancy. Physiological dormancy is caused by low growth potential of embryo, and in general, after ripening or cold stratification is useful to break seed dormancy in this case. Physical dormancy is caused by water, impermeable fruit or seed coat, and in combination of dormancy, the germination inhibited by both physical and physiological factors, and the others are morphological and morphophysiological dormancy caused by small underdeveloped embryos at dispersal. So we observed internal seed morphology by using USB microscope. As you can see in the picture, the seeds had fully developed embryos at dispersal, so they do not have morphological or morphophysiological dormancy in this case. So we can exclude morphological and morphophysiological dormancy in this classification table. Next, we checked whether the seeds have physical dormancy at dispersal. The seeds were placed on filter paper and moist with water, and then we measured the increasing seed mass for 72 hours. The seed mass increased by up to 20% according to harvest time compared to initial weight. But in general, if seed mass increases more than 20%, then we can assume that the seed coat are permeable to water, but we are not sure in this case. But when the mechanical scarification was applied, the seed mass rapidly increased by up to 6%. So in this case, it may have physical dormancy. So we can narrow the dormancy type into physical or combinational dormancy in this case. But when we consider about physical dormancy, we have to know where water can pass through. Many species with physical dormancy have water gap complexes, which are specialized areas outside of the seas. So water entry is inhibited until specific time, depending on each species. So in this case, water gap closed, but in right pictures, water gap open. Thus, water can pass through inside the seas. These water gap complexes could be organized into 3 types. Type 1, 2, 3. So type 1 water gap complex is the water gap with narrow linear openings occluded by elongated palisade cells. On the other hand, type 2 water gap complexes have narrow linear or circular openings occluded by lead-like structures formed by palisade cells. But last one, type 3 water gap complex are either circular or narrow linear openings occluded by plug-like structures, usually formed by water impermeable scalenchymous cells. So we saw the seeds in an experimental plot in an open field. Then we checked when the seeds imbibed water in the natural environment and where water can pass through inside the seeds. As you can see in the pictures, we used the dye-tracking method with safranin. In the pictures, you can see the HeLa plug-like structures was separated from the adjacent palisade cells from October. So from October, the water can imbibe water. Then water can pass through inside the water from October. But sorry, there was no germination in this period, even though the water can pass through inside the seeds. This slide shows the phenology of germination in the natural environment. The germination was delayed till April next year. But in the middle of March next year, when the temperature increased after cold during winter season, the germination increased. But we can see it took about 5 to 6 months from dispersal to germination in the natural environment. So to know the reason why the germination was delayed in nature, so we did some laboratory experiments. First, we scarified the seeds with sulfuric acid, but few seeds germinated in this case. On the other hand, cold stratification following hot water treatment or sulfuric acid treatment not only broke the seed dormancy, but also promoted germination in this case. So we can conclude that the seeds have combinational dormancy in this case. And then we treated the seeds with gibberellic acid after scarification. In this case, we found a high percentage of germination. These pictures is germinated seed. You can see the radical emerges in one side. In this case, we wonder why does seed coat open like a lid only in one side. In this case, we found that the site with emerging radical has thinner scalenchymous cells. On the other hand, the opposite side the thicker scalenchema cell than the other side. So thinner side was firstly open, then the radicle can emerge this side in the natural environment. So in conclusions, during the progress of the dormant break and germination, water gap complexes could be opened if the seeds were dispersed in autumn from mother plant, but the dispersed seed cannot germinate because the seeds need cold certification to break physiological dormancy in nature. Then the seeds can germinate in the next year, spring, because they have combination of dormancy in this case. Thank you. Thank you for your attention. Certification.
Thank you for your attention.
Thank you very much, Dr. Lee. Yeah.
Yeah. Do you
Any
hear me?
questions?
Okay.
This is amazing work, especially the details. It's a good textbook. Yeah. Any questions from the floor? Online? Now, I have a question, Dr. Lee. Thank you very much, Dr. Lee. Any questions? This is amazing work, especially the details. It's a good textbook. Any questions from the floor? Oh my gosh. Online? So, Dr. Lee, can you hear me? How about the later germination? Because I saw the plants here in US that have a lot of germination under the tree. So they're treated as a weed because there's too many germination under the tree. Do you observe anything in Korea?
In the natural environment? Your question is I saw the germinated seed in the natural environment under the trees, or if it's okay, the question again, please.
Well, I don't know if any, I just think about this nature germination to do a course. Maybe they stay in the soil too long. I think they have a lot of germination in the area. If I have one tree, then many seedlings come out.
Yeah. I often see the germinated seed in the below of the tree from the dispersed seed. I often see that.
So the same thing in Korea too?
Actually, I have not. I didn't observe the exactly about that yet. Sorry about that. Yeah.
Well, and this is probably any more questions from floor or from online? Well, if not, thank you very much, Dr. Lee.
Thank you.
And for the nice talk. Looks like we have a 30 minutes break. So hopefully everybody come back in 30 minutes. We have 4 more speakers and they conclude this sections. So I will see you in 30 minutes.
네, 마이크 테스트 하겠습니다. 마이크 테스트 하나 둘 셋 하나 둘 셋 마이크 테스트 하나 둘 셋.
Professor Dong-min, 1 minute left. We are ready here at the venue. Thank you.
1 more minute.
No, I'm sorry, my internet doesn't work in the morning, so I
Okay.
yeah, I missed this business meeting.
Good afternoon, Dong-lin Chang. Professor Dong-lin Chang here. We are ready at the venue. So could you please start to manage this session 3?
Okay.
Thank you.
Okay. Thank you. Hello, welcome back. And we have one more invited presentation and 3 more oral presentation. Our first presentation will be development of droplet vitrification procedure for the cryopreservation of aster family germplasm, and majorly authored by Heng-Hong Kim and Ho-En Ni. And Dr. Kim will be present. And Dr. Kim is a professor from Shenzhen National University with a specialty of cryopreservation of genetic resource and agrobiotechnology. He earned his PhD from Seoul National University and served as a junior research officer at the National Gene Bank and Rural Development Administration in Korea for 16 years, and the senior office for 3 more years. This means 19 years for this service. Before he returned to the academia, he worked as an APO office in Biodiversity International and FAO, which is belong to the United Nations. Dr. Kim received numerous honor and award, such as explanatory officer, and which is a excellent officer. His research include management on conservation and the utilization of genetic resource. He developed droplet vitrification protocol, use a systematic approach, and cryo banking over 1,000 garlic accessions at RDA Gene Bank of Korea. 1,000, wow, that's a lot. At Shenzhen National University, he focused on cryopreservation of endangered wild species, as well as agricultural clone germplasm. He published over 100 scientific papers, including about 70 SCI journal papers. Dr. Kim, welcome, and the floor is yours.
Professor Chang, for the kind introduction. Can you hear me? Thank you, Professor Chang, for the kind introduction. My topic is development of droplet vitrification procedure for the cryopreservation of Asteraceae composite family germplasm. Let me briefly introduce the germplasm conservation approaches using the cryopreservation. So what and how, why and how to cryopreserve the germplasm. And secondly, I'd like to introduce a systemic approach on the development of cryopreservation protocols, and then focus on the composite families. Is it not work? Okay. And then finally, cryobanking of the germplasms. As you already know, we need to conserve the germplasms of 3. 3 means the endemic and endangered and also economic plant. But I'd like to include the list, which is the clones and non-orthodox seed germplasms. among the 350,000 species of vascular plant in the world, almost 68% of these species need to be crop preserved because there are more or less 1,000 coronary germplasms and 151,000 of short-lived seed species, mostly wild relatives. And also there are 88,000 of recalcitrant seed, which means the desiccation-sensitive species. The another group may I include is the larger or largest family in the world. For example, the composite family is the largest one, almost 9%. It covers almost 9% of the whole plant kingdom. And the second is the propagation. We need to propagate to conserve and to use and even for the restoration in situ condition. Mostly I do propagate the germplasm through the in vitro culture system. So I need to introduce by the in vitro germination and then multiply it and then conserve it. That's it. And for the multiplication, in vitro multiplication, we need to simply node cutting for some species. For some other species which do not have the nodes or not rooting, and then we can induce the multiple shoot by using the BAP. And in some case, especially for the trees, we need somatic embryogenesis or organogenesis. Here you can see the composite family has been used as food. As you can see, the lettuce, chicory, and also for herbs and medicinals and some teas, and also for ornamentals, including the chrysanthemum, gobera, etc. In Korea, we have a large number of the composite family germplasms. The first one is regulated by the Ministry of Agriculture. So around the country, there are about 8 sub-gene banks which maintain the coronal composite family germplasm collections. And the total number is 1,600. A huge number of germplasms are maintained coronally in Pin gene bank. And the second one is the wild relatives, which is regulated by the government, Ministry of Environment, and also National Forest Service under the Ministry of Agriculture. Also, we have some tens of genus and 23 species of endemic and endangered species of composite family. Yeah. And it is recommended to conserve the germplasm as a complementary approach. Complementary approach means we need to conserve the germplasm in situ, but also in ex situ also. If we can conserve the seed germplasm, but also as the vegetable tissue, pollen, cell lines, and DNA, etc. And also FAO, Food and Agriculture Organization of the UN, adopted the second version of the gene bank standards for the introduction, conservation, multiplication, everything on the gene bank activities. There are 3 versions of gene bank standards. For the gene bank standard for the orthodox seed, normal seeds, and the second one is feed gene bank standards for the coronal germplasms and also some recalcitrant seed species. And the third one is gene bank standard for in vitro culture and cryopreservation. For the problem species, problem materials, it's only for the long-term conservation strategies. Yeah. As you can see, also the FAO recognized the general germplasm conservation approaches. If the seeds are orthodox, then we can just dry it and then conserve it at minus 18 degrees Celsius or at cold room temperature. But there are some species of the vegetatively propagated crops and some recalcitrant seed species we need to maintain in field gene bank or and for in vitro culture or in vitro propagation system. But those are only for the short-term or mid-term conservation strategies, and the cryopreservation is the long-term conservation strategies. In cryopreservation, we can conserve any type of materials which can be restored and also which can be tolerant to the desiccation and also tolerant to the vitrification solutions. So the seeds, foreign tissue cell, anything can be stored in liquid nitrogen. Yeah. Why cryopreservation? The cryopreservation is the sole option for the long-term conservation of problem materials of non-orthodox seed and vegetatively propagated species. So minus 130 degrees at this high, lower the temperatures, all the chemical reactions are arrested, so we can maintain the germplasm for a longer period of time with the relatively lower cost maintenance. And also there are a huge number of cases of large-scale implementation of the cryobanking for the agricultural crops, but not so many cases for the wild species yet. In Korea also we have conserved the non-orthodox seed species like ginseng seed and also for garlic and some other dormant seed species. Next, please. Yeah. Let me introduce the cryopreservation techniques. There are two classes of cryopreservation. The first one is slow cooling, simply, and the second one is rapid cooling. Slow cooling is the conventional or controlled or two-step freezing methods from the ambient temperature to minus 40 degrees Celsius very slowly. And this approach has been used for the undifferentiated cells like the suspension cells and also for the winter hardy species like fruits and some vegetables as well. And next, please. And the second one is rapid cooling. This is the main part of the cryopreservation studies. If the samples are desiccation tolerant, then we can just dry it and then freezing it, liquid nitrogen, that's all. And some cases of semi-desiccation tolerant materials, then we can apply the pre-culture with higher concentration of sucrose, then desiccate it and then cryopreserve the samples. For example, the embryonic axis of the recalcitrant seed species, we can do applying the pre-culture and then desiccate it and then cryopreserving the samples. And the second one is vitrification. This is the main part for the smaller or desiccation sensitive. Most of the vegetative tissues and recalcitrant seed species belongs to this family, these classes. And also there are so many kinds of cooling and warming devices, including the cryovials and straws and aluminum foil strips and cryoflat, loop and loop, anything else. Even for the human germplasms, they have used the sophisticated, very physical instrument for the very rapid cooling and thawing velocities. Then how to choose the best option for our material? If I have some material, then the protocol should be developed based upon the characteristics of the materials. It has been done with the trial and error basis, many cases, but it should be done based upon the characteristics of the materials. For example, the tolerant materials, if the samples are desiccant tolerant, then just desiccate it. And for sensitive materials, if there is cells, then we can apply in the vitrification rather than slow cooling. And for the shootives, we can apply in the vitrification procedures. And also, it should be selected based upon the availability of the techniques in our lab also. Yes. So the solution-based vitrification procedure is a sequencer procedure from the preparation of materials and the preconditioning or pre-culture and the ozone protection and crop protection. So ozone protection and crop protection is the main part. So I I'm going to focus on this process. And after that, cooling in liquid nitrogen and storage it. And after storage, we need to thawing them and then unloading. Unloading means the exclude or removal of the crop protectant from the samples to the solution. And then finally regrows the samples. This is the general procedures for the crop preservation of the chrysanthemum germplasm using the droplet vitrification procedure. We maintain the germplasm using the general tissue culture, using the node cuttings. And then after the final stage, we need to develop the node cuttings and then excise the shoot tips from the node cuttings. And then do preculture with a high concentration of sucrose, normally from 10% to 17%, followed by ozone protection and vitrification using the highly concentrated vitrification solutions, and then put the samples onto the aluminum foil strip, and then covered with some amount of vitrification solution, then directly cooling in liquid nitrogen, and then store in a nitrogen tank. And after storage, we need to thawing and warming the samples using the preheated unloading solution. And also, after thawing, using the same range of unloading solutions, we need unloading, removal of the crop protectants, and then just go through the regrowth phase. Yes. This is the general procedure of the solution-based vitrification procedure. We can start from the 10% sucrose, then goes up to even to 17 or 25% sucrose. Then for the ozone protection, we need another group of solutions up to 40% concentration. Then finally do crop protection with highly concentrated vitrification solutions. And then store it. And then finally goes down to the unloading solution. Then goes to the final regrowth phase. Then we can get some recovered materials. Yeah. So let me briefly introduce a systemic approach to develop the vitrification procedure. The first step is the preparation of the samples or materials. We need to develop the very healthy material, not the same as the general tissue culture method. We need to develop the very healthy, very good materials for the crop preservation. Otherwise, we cannot guarantee the higher regeneration after cryo. So it's very important. And also, it is very important to combine the general tissue culture procedures for the specific material, and then combine with the crop preservation protocol. Then we can get a successful result. Okay. The second step is preculture. Normally, we use the 10% sucrose followed by 17% sucrose. Most of the species belongs to this class, so it can be the medium and sensitive or tolerant materials. But some materials, they can tolerate to the 25% of sucrose, or some materials are very sensitive to osmotic stress, then only 10% or even only 3% is enough for this material. Yeah. And the other step is ozone protection. We also select the C435%. The solution was optimum for majority of the samples. Here are some examples of the alternative ozone protection solutions which have been developed in our group a long time ago. Here is some composition and osmolality of the solution itself. And enthalpy of the solution means the heat thermal enthalpy, which has been happened during the cooling and warming phase. We can measure the enthalpy using the differential scanning calorimetry. And then we apply to some model species, including the chrysanthemum. Here you can see C435 produced the highest regeneration after crop preservation. Yeah. The next step is crop protection, most important procedure. The important thing is the balancing of crop protection and the cytotoxicity. If we increase the concentration of the solution, then it's very helpful to crop protection, but it's also very toxic to the sample. So we need to tuning and balancing these two groups. And for the plant beautification solutions, the PBS2 and PBS3 is the most, especially the PBS2 is the most popular beautification solution, which was developed by the late Professor Sakai in Japan. I tried to develop the alternative beautification solutions. Here are some examples of the PBS2-based solutions in A3 number and PBS3-based solutions B3 numbers. Also, the characterize the solution itself using the enthalpies and the moisture content. Yeah. And then applying to the different type of materials. The first one is garlic. Yeah. Next, please. Yeah. And the second is chrysanthemum. For garlic, it is tolerant material for osmotic stress. Yeah. So mostly the PBS3 based, the right side solutions produce the higher post-crop preservation regeneration compared to PBS2 based. And the chrysanthemum is very sensitive material to osmotic stress and also very sensitive to chemical toxicity. So you can see in case of A9, this solution have higher concentration of DMSO and ethylene glycol. So we cannot get any regeneration after crop preservation even. And also some dilution of the solution worked well. Yes. Here are some examples of the applying of these alternative beautification solutions for the chrysanthemum. Here, A3 ice for 40 minutes or 55 minutes, or the B1 RT room temperature for 60 minutes produced higher regeneration after crop preservation compared to other solutions. So here are some summaries on the alternative beautification solutions. In one side, based on the cytotoxicity of the osmotic stress and chemical toxicity, whether it is tolerant or sensitive. In the other side is the size or permeability of the samples, including the tiny callus and the small meristems and the medium size of shoot tips and larger semi-permeable bulbs or rhizomes. So depending on the characteristics of the samples and the size, we can select alternative diverse options of the beautification solution for the specific materials. Here you can see in blue light, we can get higher, very higher, more than 80% of regeneration. But in the black color, we can get so-so around 60 to 80% of regeneration. But here in the red color, we cannot guarantee higher regeneration after crop preservation for these specific materials. But unfortunately, most of the samples are basically sensitive to osmotic stress, also for the chemical toxicity. Yeah. And here is some examples of the a set of treatments. We can select some number of treatments for each step and then do the experiment. Then we can figure out which condition is better for this material. Then we can formulate the standard protocol at the beginning, and then we can start the actual experiments. Here is some other examples of the, here. As you can see in chrysanthemum. Chrysanthemum is a relatively sensitive material to the chemical toxicity and also moderately sensitive to osmotic stress. For example, the PLB, protocol-like bodies of the orchids, they are very, very sensitive to both the chemical toxicity and osmotic stress. The next step is cooling in liquid nitrogen and storage it, and then rewarming the samples. And the trophic purification uses the foil strip, aluminum foil strip. It's very useful for the rapid heat transfer. So it guarantees very high rapid cooling and thawing velocity. And also the warming velocity is more important than we can get using the preheated unloading solution. Yes. Next, there is unloading, and also we can develop some alternative unloading solutions. And finally, for rigorous. As far as I examined, as far as I get some research from the wild relatives, almost all of the samples are very, very sensitive to ammonium in the post-culture media. So we need to exclude the ammonium nitrate for the initial 5 days. This is a very important step for the majority of the samples, even including the crystalline. Okay, this is the summary of the first round experiment, and the second round, after move to the university, I had some studies on the endangered wild species, and from last year, we start some other project on the crop preservation of agricultural crops, including the composite families. Here is some examples from the review on the chrysanthemum for the cold storage, including the encapsulation method and then crop preservation. And this slide shows some case studies on the wild relatives of the Danyang Ester Altaicus. This species is endemic and endangered species in Korea. And we identified the optimum pre-culture conditions 2 days with 10% sucrose, followed by 70% produced higher regeneration after cryo. And then the dilution of the butyric solution from 90 to 80% is guarantee higher regeneration after cryopreservation. And the same material with the foil guarantee higher regeneration, which means the samples are not sufficiently crop protected during the crop protection phase, because this material is very sensitive to the osmotically and cytotoxically. So we can decrease the level of crop protection treatment. And for the regrowth, only the removal of the ammonium ion in the post-culture media guarantee higher regeneration after crop preservation. As you can see, ALT3 and 4 produced higher regeneration. This slide shows the cases of the chrysanthemum variety named Yongno. From the minimum set of experiment, just composed of 5 treatments, including the standard condition. As you can see, the left side, the first one is the standard condition. But as we minor change some conditions, then we can compare which one is the critical parameter or conditions. And the last one, the right side, which is the post 2, means the including the ammonium ion in the post-culture media. Then we cannot get the higher regeneration. And last year, we cryopreserving the 40 chrysanthemum accessions. And then the average regeneration after cryo is around 70%, but some accessions produced lower regeneration after cryo. So we identified there are some problems with the subculture method. So we developed a subculture, and then the regeneration goes up again. And in this year, we also cryopreserving the material about 60 accessions. We conserve the material and regenerated in Suncheon, then transfer the samples, cryo samples, to the backup facility of the RDA gene bank. Yeah. Here are some examples of the wild relatives of the composite families. And here you can see there are some materials, they do not produce nodes. Then we can apply in the BAP to produce the multiple shoots. Yeah. Here is some other examples of the wild relatives. Here you can the similar result from the chrysanthemum and the other species also. Yeah, as a conclusion, the crowd banking has been done, is still ongoing. And also the preparation of the samples is the big issue also. And finally, there are some possible application of crop preservation. For example, the cryotherapy. Cryotherapy means the removal of the virus or pathogens, any pathogens during the crop preservation. If the crop preservation is not sufficient and successful, then most of the tissues are damaged, killed, and then only the meristems can be survived. Then we can have a chance to get the virus-free materials or pathogen-free materials. And the cryosolation also the same. If the crop protection conditions are not appropriated, then most of the cells are dead. Only the competent cell line cells can be selected or survived after cryo. Then we can have a chance to select some competent cell lines. Thank you for your attention.
Oh. Sorry, this is the automatically cut off. We do not have questions. Any questions? Let me have one question.
Yes.
Any question from floor or? If there are no more questions, thank you very, very much, Dr. Kim, and for your presentation.
Thank you.
Our next presenter is talk about genetic structure of Korean and Japanese populations of Hydrangea serrata analyzed by microsatellite. And the author are Shu Yamamoto, Yuta Abe, Tamaki Hada, Miro Kawakobo, Hirokazu Yamamoto, Pin Yong Chong, and Takashi Handa. The presenter will be Dr. Shu Yamamoto from Meiji University. Dr. Yamamoto, the floor is yours.
Okay, hello. I'm Sho Yamamoto. Today I'm going to introduce our research genetic structure of Korean Japanese population Hydrangea serrata analyzed by microsatellites. Today I'm in Japan. I attend with Zoom. Introductions. First of all, Hydrangea serrata. Hydrangea serrata is popularly used for horticulture. The inflorescence shape of this species divided into two types. In the wild type of this species, the inflorescences consist of many fertile flowers, some decorative flowers. Its names are race caps. Individuals with much decorative flower type is moped. It is rarely found in wild population. Many plants with characteristics of, such as moped, double flowers, red to blue to white, various flower colors have been selected from wild populations. These are used in horticulture purpose. such as ornamental shrubs, breeding materials. Next slide, introduction, distribution of subsection hydrangeas. Hydrangea serrata belongs to this section. Hydrangea serrata is distributed from the southern part of Korean Peninsula, Jeju Island to mainland of Japan. Hydrangea and closely related species endemic to Korea and Japan. Hydrangea maculaphila is grown in coastal area. Serrata varieties is grown in mountain snowy region. In this study, we focused Hydrangea serrata. Flower color of Hydrangea serrata. Color of decorative style flower is varied. Flower color of Hydrangea serrata is variable in Korea and west side of mainland of Japan. On the other hand, flower color of this species it is white flower from central to east area, east side of mainland of Japan. Progeny of Hydrangea serrata. Rapid markers and chloroplast DNA sequence suggested that Hydrangea serrata of Japan was separated into two groups: Western serrata groups, Eastern serrata groups. However, genetic structure of wild population is still unknown. Microsatellite or simple sequence beat markers are useful to show genetic analysis. SSR is usually 2 to 6 nucleus repeats, like this. SSR markers have developed for Hydrangea and investigated in several studies. In our previous studies, we used these SSR markers morphological analysis confirmed whether old Hydrangea cultivar imported Europe and identical with old cultivar existed in Japan. Aim of this study, there are large diversity in wild populations, such as flower colors, blue to white, and plant weight. However, genetic structure of wild population is still unknown. In this study, comparing the Korean and Japanese Hydrangea serrata, we conduct DNA analysis by nuclear SSR markers. Materials. 40 individuals from Jeju Island of Korea was used. In Japan, we used 3 population of western areas, 5 population of central areas, 5 population of northwestern area, and 6 population of eastern area was used. 188 individuals from 19 population of Japan was used for analysis. DNA was isolated from fresh or dried leaves by the Sweetan methods and start at -30 until use. In the microsatellite analysis, genotypes of each sample are scored by using 6 pairs of SSR markers. The size of PCR products was determined peak scanner software after electrophoresis genetic analyzer. In the data analysis, during the principal coordinate analysis, PCOS based on net genetic distance was performed using GenRx. And also split network was used in this study. Next slide is result and discussion. It is a result of PCOA principal coordinate analysis. This analysis suggested that the sample collected from the population in Jeju Island of Korea, it had closely related to genetic structure to that closely related to sample collected from the population in the western area of Japan. And the Korean and western area population of hydrangea serrata has various color flowers. And analysis of chloroplast map-K and RBCL sequence revealed the Korean and western area of Japan individuals of hydrangea serrata form monophyletic groups. So our results supported previous studies. Next slide is result of split network. The split network shows similar result of PCOS. The population collected in the Jeju of Korea is, and western area population was closely related to northwestern area populations. And eastern area population and central area population clearly separated from other area populations. Next slide, discussions. So in the study, population spread northwestern areas populations. The population northwestern population may spread from the western to northwestern area. And the central area population were genetically different from western and northwestern populations. So it may, mountain of central area may prevent the gene flow among these areas. The analysis also revealed the genetic difference among eastern, central, and the other area populations. Other studies, the rapid and chloroplast analysis suggested that hydrangea serrata of Japan was separated into western serrata group, which has blue flower group, blue to barrier blue flower groups, eastern area groups, which has white flower groups. So other studies suggested Sisca Mountain is one of the border of this species. However, in these studies, SSR analysis suggested clear differences among eastern area and central areas. So there are other genetic border was suggested. Fossa Mogna is known as a frostic region. And it's in this area. So this area is known as frostic region, and it is rich in endemic plants. The distribution of hydrangea serrata is not related to frostic regions. However, genetic differentiation may be related to it. In this study, the result of this study suggests genetic diversity in white population of hydrangea serrata. This species has large diversity and genetic differentiation among wild populations. So This species have been used for breeding cultures. However, there are few studies on wild population genetic diversity. The present study is first investigated SSR analysis of wild population of Hydrangeas acerata. The information difference among wild population will be useful information of stratificating parents individuals for breeding programs. Thank you for your attention.
Thank you very much. Any questions for Dr. Yamamoto? Any questions from floor or from online?
Yes, I have a question.
Good.
White spaces, different distribution area, which divided by ISR markers. I was wondering, is that among the different area, is there some morphological or ecological characteristics can be divided by SSR or the distribution? That means what's the difference between central area, eastern area, or north area in morphological differences?
So now I compared morphological difference. Central area population and eastern area populations, flower color is white, but plant shape and leaf shape is little different among populations.
You mean the flower color is different between different area?
Yeah.
Okay, that's very interesting.
Thank you.
There are great genetic diversity among one species. Yeah, okay, thank you.
Thank you very much, Dr. Yamamoto. Let me conclude this presentation, continue due to the timing. Our next topic will be distribution and the phylogeography of nature populations of Chinese white plum, Prunus salacina in Korea. And this is authored by Jimin Zhang, Hoon Kim, Chung Hao Hu, Chen-Ying Kim, and Xiangyang Kim. Dr. Jimin Zhang from Korea Relation Arboretum will present this topic. Dr. Zhang, is yours.
Hi everyone, I am Jae-min Jung from Korea National Arboretum. What I will be presenting is the distribution and phylogeography of natural populations of Chinese wild plum, Prunus salacina in Korea. Next slide.
Jae-min Zhang from Korea Relation Arboretum.
Genus Prunus contains around 200 texts worldwide. Prunus salacina is also called Chinese or Oriental wild plum. This species is distributed in central, eastern, and northern regions of China. And it's an important genetic resource for plum tree improvement. As you can see on the map, Prunus salacina is distributed in central, eastern, and northern regions of China. But in South Korea, some natural populations of Prunus salacina were found in 2016. Some small populations were distributed in central regions of the Korean peninsula, especially in Dimjeet and the surrounding area. For the use of tree improvement and cultivation on natural populations of Prunus salacina in Korea, the purpose of this study was to survey the distribution area and ecological characteristics, and to investigate the taxonomic and the phylogenetic relationship between related taxa. For survey of the distribution area, distribution area of the natural populations had surveyed in flowering season, and surveyed ecological characteristics, number and size of subpopulations, height and deviation of tree, size of fruit and seed had surveyed. For phylogenetic study, samples were collected 13 accessions, including 6 accessions in China. For DNA sequencing, 5 genes were selected in nuclear DNA and cpDNA. This is a distribution area map of Prunus in Korea. Blue dots indicate regions collected in Ganggae-gun of Jagangdo, North Korea in 1910 and 1911. Black dots indicate regions found in Yanggu-gun, Injae-gun, and Goseong-gun of Gangwon-do in 1916. These photographs show the natural habitats of flowers and fruits of Prunus sericina in Korea. This is the result of distribution of DVH class at the natural populations of Prunus sericina in Korea in the range of 2 to 18 centimeter. The proportion of the 20 centimeter DVH tree was the highest. This is the result of distribution of height class at the natural populations of Prunus sericina in Korea. In the range of 8 meter high, the proportion of the 6 meter tall tree was the highest. This table shows the size of the fruit and the seed in Prunus sericina. Fruit is very small, less than 2 centimeters in size. Red colored fruits have grown in sunshine, and greenish yellow fruits on the shade. This table shows the length of sequence for 5 markers from 13 samples related to Prunus sericina. For the 5 markers, base pair of sequence ranges from 600 A3 base pair to 1112 base pair. This figure shows maximum likelihood tree of 13 samples based on the nuclear ITS. In nuclear ITS gene, 5 samples of Korea and 6 of China were genetically identical. Figure 6 shows maximum likelihood tree of 13 samples based on the 4 cpDNA regions. In 4 cpDNA regions, 5 samples of Korea and 6 of China were also genetically identical. In conclusion, the samples collected in Korea were same species as Prunus sericina of China, and morphological characters such as leaf flower and fruits were also same as Prunus sericina of China. Thank you for your attention. Prunus sericina. For your attention.
Thank you. And we have time for questions. Any question from the floor? Or any question from online about those? Dr. John, is this fruit in Korea people collect it and eat it? What is the fruit for?
Yes, I think it's a fruit. It's a very small and.
Thank you, and we have time for questions. Any question from the floor? Well, if a low question, let me thank you, Dr. John, for his presentation. And we will continue to go to the next one. The last one is also the best one will be for today's sections. And the next topic will be floral analysis of shrub plants in 32 Hunan nature parks and its potential application on gardening. This is authored by Liu Qingyang, Yin Liu, Yupeng Zhang, and Fei Yong Liao. Professor Liu Qingyang will be present, and Professor Liu Qingyang is from Central South University of Forestry and Technology. Liu Qin, it's all yours.
Thank you very much for Dr. Donglin Zhang. It's my honor to speak here, and thanks for giving me this opportunity to share my research with you. And today, I'd like to talk about floral analysis and the application prospects of wild shrubs in Hunan province. My presentation has 4 parts. I'll start with a brief introduction of my research context. And one of my purpose of this research is that I hope to find some beautiful shrubs from the national parks that can be used in wall greening. And here is part of my research base of wall greening. This research is conducted in Hunan province, which is located in central south of China, which is in the middle reaches of the Yangtze River. And the landform is dominated by mountains and hills. Here is a map of Hunan province. The western part of Hunan are mainly mountains and peaks, which the eastern part of Hunan are basins, plains, and hills. The 22 national parks in this study is evenly distributed in Hunan. Here is a list of 22 national parks in Hunan province. And literature research and field survey were used to collect the shrub data, and followed by floral analysis. A non-investigation and non-transact survey were used to create a plant inventory of wild shrubs. B, for the species that cannot be identified on site, the plant specimens were took back to lab and compared with photos from books and official online taxonomy based database, such as flora of China and the flora of Hunan. Then we screened and classified the wild shrub inventory based on their aesthetic value and applicability in landscape. We identified 1,135 species of wild shrubs in the 32 national parks. And the result of the floral analysis shows that they belong to 94 families. Among them, 9 species are gymnosperms, belong to 4 families, and 1,126 species are angiosperms, belong to 90 families. Among the 90 families of angiosperms, there are 5 classes according to the number of species in the plant families. The largest family contains more than 50 species, and the second largest family contains 30 species to 49 species. The third largest family contains 11 species and to 29 species. The fourth largest family contains 2 species to 10 species, and the last one, single species family. Here is a table, genus and species in the family of shrub angiosperms in 32 parks. Among the 284 angela of angiosperm shrubs, there are 3 classes or according to the number of species in the plant genera. The largest genera contain more than 20 species, and the second largest genera contain 10 species to 19 species. The third largest genera contain less than 10 species. Here is a table about genera and species composition of shrub angiosperms in the 32 parks. The distribution patterns of families according to Wu Zhongyi 2003, the area types of the world families of seed plants. The area types of shrub angiosperms families are classified into 10 area types and 12 subtypes in those parks. Here is a table about the distribution types. Distribution patterns of genera. According to Wu Zhongyi 1991, the area types of Chinese genera of seed plants, the area types of shrub angiosperms genera are classified into 14 area types and 14 subtypes in those parks. This is a table about the distribution patterns of shrub angiosperms genera in 32 parks. Based on the aesthetic value of shrubs, we screened out 660 species of plants with landscape application potential and divided them into 4 types. The first one is foliage shrubs, and the second one is flowering shrubs. The third one is fruiting shrubs, and the last one is other shrubs. First one, foliage shrubs. There are 35 families, 80 genera, and 98 species of foliage shrubs with a unique leaf shape or bright leaf color. Here is there are some beautiful pictures about foliage shrubs. Here also the foliage shrubs. And the second is flowering shrubs. There are 72 families, 125 genera, and 328 species of flowering shrubs with distinctive colors, large amounts of flowers, and beautiful shape. Representative families, genera, and species are listed here. Very beautiful flowers. And the third one, fruiting shrubs. There are 46 families, 93 genera, and 227 species of fruiting shrubs with varying colored and large amount of fruits in different seasons. Representative families, genera, and species are listed here. There are some very beautiful fruits. And the fourth one, other shrubs, contain 5 families, 7 genera, and 7 species. That's all for my presentation. Thank you very much.
Okay, thank you, Professor Yang. And Hunan is definitely have a very rich flora and have a great potential for ornamental plants for your landscape. So this is Dr. Liu Qingyang have analyzed. Hopefully someday you can travel in there and look at more plants. Now, any questions? We have time for questions. Any question from floor or from online or together?
Any questions from floor?
No questions? Well, Liu Qin, how many plants have you used for your landscape?
So far, maybe 20, more than 20 plants from the natural parks we collect.
You collected and bring to use in the landscape?
Yes.
Okay.
More than 20.
This is great. So you begin to use our nature species. This is excellent because we do need to use more. Any more questions from online? Well, if not, thank you, Dr. Liu Qingyang, for your presentation. And this will conclude our section 3 about the conservation, plant conservation, gene plasma conservation. And I would like to take this opportunity to thank all of you for your participation. And also, I greatly appreciate the organizer and for this wonderful symposium and bring us together. And let's get a round of applause for the organizer, for the speaker, for all of us. Thank you very much.
Thank you very much, Donglin Zhang, for your wonderful leading for session 3. And thank you for all the speakers for the session 3. It was a very special experience for us to have Dr. Donglin Zhang as a chair with online. So thank you. And now we have lunchtime, and for online participants, we are preparing the photo exhibition and video clip by Korean National Arboretum during lunchtime, like yesterday. And after that, we will have poster session continuously. So please join the e-poster session through the website until 2:30. There will be our last session, session 4, Applicants of Modern Technology. And we have keynote and invited speakers by Professor Ki Sang Zhang and Sang Yong Kim for session 4. So please enjoy your lunchtime and the poster session. And I would like to have you again at 2:30. Thank you.
Where the present and past coexist. Korea National Arboretum. While everyone is busy chasing today, the forest is changing at a slow pace. A secretive 550 years. The start of which can be found at the Royal Forest in Gwangnam, where the tomb of King Sejong, the 7th king of the Joseon dynasty, and Queen Jeonghye are located. The Royal Forest has embraced the tomb over time. From the nation's experimental forest dedicated to research and study of forestry, to an arboretum researching vegetation and forest today. The forest changes every day, telling us many different things. Meeting diverse plant life over 4 seasons. Studying the bountiful gifts of nature. A glimpse into the precious life of nature's monuments and hidden existence of rare species can be found here. The forest has become fertile and its value heightened. Designated by UNESCO in 2010 as a biosphere reserve, the nation's largest experimental forest research center. The nation's only research institute dedicated to the diverse species of the forest. Our nation's representative forest ecosystem report. It has become a resting haven for citizens, where the environment and culture harmonize as one. Think about the tomorrow that the forest brings us. A forest of healing that we seek out in times of need. A forest of hope to find the sounds of nature. A forest to bring us back to the beginning, to cherish nature's natural beauty. A treasured forest for our tomorrow. A space which is shared by the present and the past. Dreaming of a beautiful tomorrow, living in coexistence. We look forward to making a 1,000-year history at Korea National Arboretum.
Forests have endured a time beyond people's imaginations. Forests have numerous stories to tell. They are the habitat of countless lives and a place of rejuvenation for people. Let's meet the people who are protecting the lives in the forests. In 1999, the Korea National Arboretum was established to collect, preserve, and study the life forms of forests in a systematic manner. The Korea National Arboretum has organized specialized gardens which are open to the general public and experts by their use and taxonomical characteristics. The Korea National Arboretum also consists of specialized facilities for specialized activities. The Korea National Herbarium is the nation's first exclusive specimen facility, established to contribute to further research in related sectors through procuring large numbers of specimens, establishing accurate species categorization systems, and enhancing the usage of specimens through impeccable management and conservation. The seed bank is a mandatory facility for long-term preservation of the seeds, which are essential to the research, conservation, and usage of the plant resources. It is equipped with a high-tech management system for long-term preservation of seeds. The Tropical Plant Resources Research Center was established in 2009 in an effort to expand the scope of forestry research. It will allow us to prepare for environmental changes and heighten the value of plant resources. The Forest Museum is a place that exhibits the history and future of Korea's forests and forestry. It features various exhibits related to forestry, relics, and woodwork. The research team is climbing mountains to study the forest organisms of the Korean Peninsula. Monitoring the habitat on a regular basis is a crucial part of protecting our natural environment. The insect team and mushroom team are also collecting specimens. Insects and mushrooms are key constituents of the forests, and there is an extensive research regarding them. Let's meet the preservation team, who are working to protect Korea's endemic plants. Observing the habitat of plants and protecting endangered species are two of the Korea National Arboretum's key roles. Reviving endangered plants is also very important. Each year, the Korea National Arboretum designates endangered rare plants and restores them in a systematic manner. Since we can't bring back nature once it's lost, the Korea National Arboretum's efforts to protect this nature must continue. Preserving plants that represent each region is one of the Korea National Arboretum's important roles. Preservation of such plants is crucial in determining and researching the habitat and environment of Korea's endemic and rare plants. Proliferating endangered plants, restoring and improving them, the Korea National Arboretum is working to unleash the full potential value of the plants. By forming the DMZ National Arboretum to preserve the forest ecosystem of the DMZ, the Korea National Arboretum is securing the biodiversity of North Korea's plants and is studying rare, endemic, and endangered species. Through systematic preservation activities and exchanges with arboretums and botanical gardens overseas, the Korea National Arboretum is leading efforts to study forest organisms around the world and discovering their value. The Korea National Arboretum is hosting a daily gardening competition to expand the gardening culture in everyday life and supporting the development of gardens in schools using Korean wildflowers. Furthermore, it is working to increase global awareness of Korean gardens. The forest is an open-air learning site of life. They learn about the various organisms that inhabit the forest, as well as interesting tales about the forest. The BioBlitz, a biodiversity exploration competition, is a great opportunity to learn about all the organisms in the forest, including plants, animals, insects, and reptiles. It is a time when people can interact with the forest and the organisms in it. After the hustle and bustle, the day is over at the Arboretum, but the meaningful efforts of the Korea National Arboretum will continue throughout the year. Korea's leading forest organism research institute, Korea National Arboretum. Based on its outstanding achievements, it is now preparing to soar to new heights. By improving its collection, research, and preservation of forest resources, and expanding the scope of forest research, Korea National Arboretum will establish itself as a world-class arboretum. Korea National Arboretum. Forests have endured a time beyond people's imaginations. Forests have numerous stories to tell. They are the habitat of countless lives and a place of rejuvenation for people. Let's meet the people who are protecting the lives in the forests. Kwangneung Forest, where the Korea National Arboretum is located, has been protected as a royal forest since 540 years ago. Possessing a stunning natural environment, it is the habitat of life, ranging from rare plants to natural monuments. It's why the value of Kwangneung Forest was recognized globally and designated as a UNESCO biosphere. Kwangneung Forest is a natural masterpiece with a harmony of life, presenting unique and stunning scenery in each of the 4 seasons. It's a new morning at the Arboretum. The Korea National Arboretum is a special place where you can see the Kwangneung Forest and the various organisms that inhabit it. In 1999, the Korea National Arboretum was established to collect, preserve, and study the life forms of forests in a systematic manner. Today, it studies plants and insects that inhabit the forests, researches seeds and specimens, organizes exhibits, and provides forestry education. The Korea National Arboretum has organized specialized gardens which are open to the general public and experts by their use and taxonomical characteristics. The specialized gardens include the Ornamental Tree Garden, presenting a collection of trees with high ornamental value. The Flowering Tree Garden, which exhibits trees with beautiful flowers. And the Aquatic Plant Garden, Medicinal Plant Garden, Rare and Endemic Plant Archive, and Temperate House. The Korea National Arboretum also consists of specialized facilities for specialized activities. The Korea National Herbarium is the nation's first exclusive specimen facility established to contribute to further research in related sectors through procuring large numbers of specimens, establishing accurate species categorization systems, and enhancing the usage of specimens through impeccable management and conservation. The seed bank is a mandatory facility for long-term preservation of the seeds, which are essential to the research, conservation, and usage of the plant resources. It is equipped with a high-tech management system for long-term preservation of seeds. The Tropical Plant Resources Research Center was established in 2009 in an effort to expand the scope of forestry research. It contributes the collection and preservation of tropical plant resources of infinite value, and to secure future resources which will allow us to prepare for environmental changes and heighten the value of plant resources. The Forest Museum is a place that exhibits the history and future of Korea's forests and forestry. It features various exhibits related to forestry, relics, and woodwork. The research team is climbing mountains to study the forest organisms of the Korean Peninsula. Today, the team is here to conduct an extensive study of the organisms that inhabit the Paektu Mountains. The researchers observe each flower and tree, recording their number and characteristics. Monitoring the habitat on a regular basis is a crucial part of protecting our natural environment. The insect team and mushroom team are also collecting specimens. Insects and mushrooms are key constituents of the forests, and there is an extensive research regarding them. The efforts to protect the beautiful natural environment and biodiversity must continue until we complete the biological survey of the Korean Peninsula. Let's meet the preservation team, who are working to protect Korea's endemic plants. The Korea National Arboretum is leading efforts to preserve life forms throughout Korea. Observing the habitat of plants and protecting endangered species are two of the Korea National Arboretum's key roles. Reviving endangered plants is also very important. Recently, the Korea National Arboretum successfully revived a rare orchid through proliferation. Each year, the Korea National Arboretum designates endangered rare plants and restores them in a systematic manner. Throughout the country, there are restoration sites that have been restored by the Korea National Arboretum. Since we can't bring back nature once it's lost, the Korea National Arboretum's efforts to protect this nature must continue. Forest organisms collected throughout the country are brought to the Korea National Arboretum. Creating specimens that preserve the natural forms of forests and studying the insect ecology are crucial steps in securing important biological resources. The Korea National Arboretum is recognized as the base camp of Korea's biological resources for its systematic management of specimens and plant seeds. Through this effort, the Korea National Arboretum builds a world-class biological resources knowledge base of organisms in Korea. Preserving plants that represent each region is one of the Korea National Arboretum's important roles. Preservation of such plants is crucial in determining and researching the habitat and environment of Korea's endemic and rare plants. Discovering the potentialities of plant resources is crucial for our future. Proliferating endangered plants, restoring and improving them, the Korea National Arboretum is working to unleash the full potential value of the plants. It is also discovering the infinite power of the plant genes. By forming the DMZ National Arboretum to to preserve the forest ecosystem of the DMZ, the Korea National Arboretum is securing the biodiversity of North Korea's plants and is studying rare, endemic, and endangered species. The Gwangnam Forest is a UNESCO biosphere, which means that its value is recognized by the world. Through systematic preservation activities and exchanges with arboretums and botanical gardens overseas, the Korea National Arboretum is leading efforts to study forest organisms around the world and discovering their value. The Korea National Arboretum is hosting a daily gardening competition to expand the gardening culture in everyday life and supporting the development of gardens in schools using Korean wildflowers. Furthermore, it is working to increase global awareness of Korean gardens. Today, we have delightful guests at the Korea National Arboretum. The Forest Museum is a popular learning site as you can see all the plants of Gwangnam Forest in one place. It is considered the best place to learn about and to understand the forest. The forest is an open-air learning site of life. As visitors touch and experience the forest hands-on, they learn about the various organisms that inhabit the forest, as well as interesting tales about the forest. The hands-on experience in the forest offers learning for everyone, regardless of age. The BioBlitz, a biodiversity exploration competition, is a great opportunity to learn about all the organisms in the forest, including plants, animals, insects, and reptiles. It is a time when people can interact with the forest and the organisms in it. After the hustle and bustle, the day is over at the Arboretum, but the meaningful efforts of the Korea National Arboretum will continue throughout the year. Korea's leading forest organism research institute, Korea National Arboretum. Based on its outstanding achievements, it is now preparing to soar to new heights. By improving its collection, research and preservation of forest resources, and expanding the scope of forest research, Korea National Arboretum will establish itself as a world-class arboretum. Korea National Arboretum.
where the present and past coexist. Korea National Arboretum. While everyone is busy chasing today, the forest is changing at a slow pace. A secretive 550 years. The start of which can be found at the Royal Forest in Gwangnam, where the tomb of King Sejong, the 7th king of the Joseon dynasty, and Queen Jeonghye are located. The Royal Forest has embraced the tomb over time. From the nation's experimental forest dedicated to research and study of forestry, to an arboretum researching vegetation and forest today. The forest changes every day, telling us many different things. Meeting diverse plant life over 4 seasons. Studying the bountiful gifts of nature. A glimpse into the precious life of nature's monuments and hidden existence of rare species can be found here. The forest has become fertile and its value heightened. Designated by UNESCO in 2010 as a biosphere reserve, the nation's largest experimental forest research center, the nation's only research institute dedicated to the diverse species of the forest, our nation's representative forest ecosystem report. It has become a resting haven for citizens, where the environment and culture harmonize as one. Think about the tomorrow that the forest brings us. A forest of healing that we seek out in times of need. A forest of hope to find the sounds of nature. A forest to bring us back to the beginning, to cherish nature's natural beauty. A treasured forest for our tomorrow. A space which is shared by the present and the past. Dreaming of a beautiful tomorrow, living in coexistence. We look forward to making a 1,000-year history at Korea National Arboretum.
Forests have endured a time beyond people's imaginations. Forests have numerous stories to tell. They are the habitat of countless lives and a place of rejuvenation for people. Let's meet the people who are protecting the lives in the forests. In 1999, the Korea National Arboretum was established to collect, preserve, and study the life forms of forests in a systematic manner. The Korea National Arboretum has organized specialized gardens which are open to the general public and experts by their use and taxonomical characteristics. The Korea National Arboretum also consists of specialized facilities for specialized activities. The Korea National Herbarium is the nation's first exclusive specimen facility, established to contribute to further research in related sectors through procuring large numbers of specimens, establishing accurate species categorization systems, and enhancing the usage of specimens through impeccable management and conservation. The seed bank is a mandatory facility for long-term preservation of the seeds, which are essential to the research, conservation, and usage of the plant resources. It is equipped with a high-tech management system for long-term preservation of seeds. The Tropical Plant Resources Research Center was established in 2009 in an effort to expand the scope of forestry research. It will allow us to prepare for environmental changes and heighten the value of plant resources. The Forest Museum is a place that exhibits the history and future of Korea's forests and forestry. It features various exhibits related to forestry, relics, and woodwork. The research team is climbing mountains to study the forest organisms of the Korean Peninsula. Monitoring the habitat on a regular basis is a crucial part of protecting our natural environment. The insect team and mushroom team are also collecting specimens. Insects and mushrooms are key constituents of the forests, and there is an extensive research regarding them. Let's meet the preservation team, who are working to protect Korea's endemic plants. Observing the habitat of plants and protecting endangered species are two of the Korea National Arboretum's key roles. Reviving endangered plants is also very important. Each year, the Korea National Arboretum designates endangered rare plants and restores them in a systematic manner. Since we can't bring back nature once it's lost, the Korea National Arboretum's efforts to protect this nature must continue. Preserving plants that represent each region is one of the Korea National Arboretum's important roles. Preservation of such plants is crucial in determining and researching the habitat and environment of Korea's endemic and rare plants. Proliferating endangered plants, restoring and improving them, the Korea National Arboretum is working to unleash the full potential value of the plants. By forming the DMZ National Arboretum to preserve the forest ecosystem of the DMZ, the Korea National Arboretum is securing the biodiversity of North Korea's plants and is studying rare, endemic, and endangered species. Through systematic preservation activities and exchanges with arboretums and botanical gardens overseas, the Korea National Arboretum is leading efforts to study forest organisms around the world and discovering their value. The Korea National Arboretum is hosting a daily gardening competition to expand the gardening culture in everyday life and supporting the development of gardens in schools using Korean wildflowers. Furthermore, it is working to increase global awareness of Korean gardens. The forest is an open-air learning site of life. They learn about the various organisms that inhabit the forest, as well as interesting tales about the forest. The BioBlitz, a biodiversity exploration competition, is a great opportunity to learn about all the organisms in the forest, including plants, animals, insects, and reptiles. It is a time when people can interact with the forest and the organisms in it. After the hustle and bustle, the day is over at the Arboretum, but the meaningful efforts of the Korea National Arboretum will continue throughout the year. Korea's leading forest organism research institute, Korea National Arboretum. Based on its outstanding achievements, it is now preparing to soar to new heights. By improving its collection, research and preservation of forest resources, and expanding the scope of forest research, Korea National Arboretum will establish itself as a world-class arboretum. Korea National Arboretum. Forests have endured a time beyond people's imaginations. Forests have numerous stories to tell. They are the habitat of countless lives and a place of rejuvenation for people. Let's meet the people who are protecting the lives in the forests. Gwangnam Forest, where the Korea National Arboretum is located, has been protected as a royal forest since 540 years ago. Possessing a stunning natural environment, it is the habitat of life, ranging from rare plants to natural monuments. It's why the value of Gwangnam Forest was recognized globally and designated as a UNESCO biosphere. Gwangnam Forest is a natural masterpiece with a harmony of life, presenting unique and stunning scenery in each of the 4 seasons. It's a new morning at the Arboretum. The Korea National Arboretum is a special place where you can see the Gwangnam Forest and the various organisms that inhabit it. In 1999, the Korea National Arboretum was established to collect, preserve, and study the life forms of forests in a systematic manner. Today, it studies plants and insects that inhabit the forests, researches seeds and specimens, organizes exhibits, and provides forestry education. The Korea National Arboretum has organized specialized gardens which are open to the general public and experts by their use and taxonomical characteristics. The specialized gardens include the Ornamental Tree Garden, presenting a collection of trees with high ornamental value. The Flowering Tree Garden, which exhibits trees with beautiful flowers. And the Aquatic Plant Garden, Medicinal Plant Garden, Rare and Endemic Plant Archive, and Temperate House. The Korea National Arboretum also consists of specialized facilities for specialized activities. The Korea National Herbarium is the nation's first exclusive specimen facility established to contribute to further research in related sectors through procuring large numbers of specimens, establishing accurate species categorization systems, and enhancing the usage of specimens through impeccable management and conservation. The seed bank is a mandatory facility for long-term preservation of the seeds, which are essential to the research, conservation, and usage of the plant resources. It is equipped with a high-tech management system for long-term preservation of seeds. The Tropical Plant Resources Research Center was established in 2009 in an effort to expand the scope of forestry research. It contributes the collection and preservation of tropical plant resources of infinite value and to secure future resources which will allow us to prepare for environmental changes and heighten the value of plant resources. The Forest Museum is a place that exhibits the history and future of Korea's forests and forestry. It features various exhibits related to forestry, relics, and woodwork. The research team is climbing mountains to study the forest organisms of the Korean Peninsula. Today, the team is here to conduct an extensive study of the organisms that inhabit the Paektu Mountains. The researchers observe each flower and tree, recording their number and characteristics. Monitoring the habitat on a regular basis is a crucial part of protecting our natural environment. The insect team and mushroom team are also collecting specimens. Insects and mushrooms are key constituents of the forests, and there is an extensive research regarding them. The efforts to protect the beautiful natural environment and biodiversity must continue until we complete the biological survey of the Korean Peninsula. Let's meet the preservation team who are working to protect Korea's endemic plants. The Korea National Arboretum is leading efforts to preserve life forms throughout Korea. Observing the habitat of plants and protecting endangered species are two of the Korea National Arboretum's key roles. Reviving endangered plants is also very important. Recently, the Korea National Arboretum successfully revived a rare orchid through proliferation. Each year, the Korea National Arboretum designates endangered rare plants and restores them in a systematic manner. Throughout the country, there are restoration sites that have been restored by the Korea National Arboretum. Since we can't bring back nature once it's lost, the Korea National Arboretum's efforts to protect this nature must continue. forest organisms collected throughout the country are brought to the Korea National Arboretum. Creating specimens that preserve the natural forms of forests and studying the insect ecology are crucial steps in securing important biological resources. The Korea National Arboretum is recognized as the base camp of Korea's biological resources for its systematic management of specimens and plant seeds. Through this effort, the Korea National Arboretum builds a world-class biological resources knowledge base of organisms in Korea. Preserving plants that represent each region is one of the Korea National Arboretum's important roles. Preservation of such plants is crucial in determining and researching the habitat and environment of Korea's endemic and rare plants. Discovering the potentialities of plant resources is crucial for our future. Proliferating endangered plants, restoring and improving them, the Korea National Arboretum is working to unleash the full potential value of the plants. It is also discovering the infinite power of the plant genes. By forming the DMZ National Arboretum to preserve the forest ecosystem of the DMZ, the Korea National Arboretum is securing the biodiversity of North Korea's plants and is studying rare, endemic, and endangered species. The Gwangnam Forest is a UNESCO biosphere, which means that its value is recognized by the world. Through systematic preservation activities and exchanges with arboretums and botanical gardens overseas, the Korea National Arboretum is leading efforts to study forest organisms around the world and discovering their value. The Korea National Arboretum is hosting a daily gardening competition to expand the gardening culture in everyday life and supporting the development of gardens in schools using Korean wildflowers. Furthermore, it is working to increase global awareness of Korean gardens. Today, we have delightful guests at the Korea National Arboretum. The Forest Museum is a popular learning site as you can see all the plants of Gwangnam Forest in one place. It is considered the best place to learn about and to understand the forest. The forest is an open-air learning site of life. As visitors touch and experience the forest hands-on, they learn about the various organisms that inhabit the forest, as well as interesting tales about the forest. The hands-on experience in the forest offers learning for everyone, regardless of age. The BioBlitz, a biodiversity exploration competition, is a great opportunity to learn about all the organisms in the forest, including plants, animals, insects, and reptiles. It is a time when people can interact with the forest and the organisms in it. After the hustle and bustle, The day is over at the Arboretum, but the meaningful efforts of the Korea National Arboretum will continue throughout the year. Korea's leading forest organism research institute, Korea National Arboretum. Based on its outstanding achievements, it is now preparing to soar to new heights. By improving its collection, research and preservation of forest resources, and expanding the scope of forest research, Korea National Arboretum will establish itself as a world-class arboretum. Korea National Arboretum.
Where the present and past coexist. Korea National Arboretum. While everyone is busy chasing today, the forest is changing at a slow pace. A secretive 550 years. The start of which can be found at the Royal Forest in Gwangnam, where the tomb of King Sejong, the 7th king of the Joseon dynasty, and Queen Jeonghye are located. The Royal Forest has embraced the tomb over time. From the nation's experimental forest dedicated to research and study of forestry, to an arboretum researching vegetation and forest today. The forest changes every day, telling us many different things. Meeting diverse plant life over 4 seasons. Studying the bountiful gifts of nature. A glimpse into the precious life of nature's monuments and hidden existence of rare species can be found here. The forest has become fertile and its value heightened. Designated by UNESCO in 2010 as a biosphere reserve, the nation's largest experimental forest research center, the nation's only research institute dedicated to the diverse species of the forest, our nation's representative forest ecosystem report. It has become a resting haven for citizens, where the environment and culture harmonize as one. Think about the tomorrow that the forest brings us. A forest of healing that we seek out in times of need. A forest of hope to find the sounds of nature. A forest to bring us back to the beginning, to cherish nature's natural beauty. A treasured forest for our tomorrow. A space which is shared by the present and the past. Dreaming of a beautiful tomorrow, living in coexistence. We look forward to making a 1,000-year history at Korea National Arboretum.
Forests have endured a time beyond people's imaginations. Forests have numerous stories to tell. They are the habitat of countless lives and a place of rejuvenation for people. Let's meet the people who are protecting the lives in the forests. In 1999, the Korea National Arboretum was established to collect, preserve, and study the life forms of forests in a systematic manner. The Korea National Arboretum has organized specialized gardens which are open to the general public and experts by their use and taxonomical characteristics. The Korea National Arboretum also consists of specialized facilities for specialized activities. The Korea National Herbarium is the nation's first exclusive specimen facility, established to contribute to further research in related sectors through procuring large numbers of specimens, establishing accurate species categorization systems, and enhancing the usage of specimens through impeccable management and conservation. The seed bank is a mandatory facility for long-term preservation of the seeds, which are essential to the research, conservation, and usage of the plant resources. It is equipped with a high-tech management system.
Good afternoon, everyone. Have you ever enjoyed the lunchtime? Now we are preparing for session 4, application of modern technology. So the chair of this session is Professor Ki-byung Lim from Gyeongbuk National University, Korea. We are preparing for 5 interesting speech again for this session. So please enjoy the session with Ki-byung Lim. Thank you. He is waiting for me too right now. So now we are connecting to our online system. Okay.
Good afternoon. My name is Ki-byung Lim from Gyeongbuk National University. I'm very honored to be as a chairman of the session 4, application of modern technology for the ISGO 2020, the International Symposium on Germplasm of Ornamentals. Okay. This session is last session of this symposium. We are quite the in-depth discussion and presentation. Those are ready from now. Actually, we have one keynote presentation and one invited presentation, and other 3 oral presentation will be continued. So now it's 2:30 East Asia hour. New York is now 1:30, very early morning, and Sydney 4:30 in the afternoon. So first keynote presentation will be given by Professor Ki-byung Jang. Professor Jang Ki-byung started his study in Beijing Forest University in 1977, and he was promoted to professor and doctoral supervisor in 1993. He is now vice president of Beijing Forest University, vice president of Chinese Society for Horticultural Science, and director of Ornamental Horticultural Committee of Chinese Society for Horticultural Science. So, Professor Ki-byung Jang, are you now ready? Yes. Let's give him a big hand. Could you please share your PPT file? Could you check your audio? Yes, thank you. Professor Zhang, would you start your presentation? Yeah, you can start. Actually, you have 1 hour, but I hear that you can, your presentation will be about 30 minutes or more or less. So are you ready? Professor Zhang, do you hear me?
Yes.
So you can start now.
Okay.
Yeah.
Okay. Good afternoon, ladies and gentlemen. It's my pleasure to be able to attend the online conference and have a chance to share my studies and experiences with all of you here. Before my report, I would like to thank Professor Byung-Yung Chung, the convener of this conference, for his effort in organizing the international academic events on occasion of global coronavirus epidemic. He and his team overcome a lot of difficulties and made the symposium hold on time. So what I'm going to talk today is analysis of characteristic aroma components of prunus movement based on genomics and cold resistance breeding. First, I'd like to give a brief introduction about what is May or what are the characteristics of Mayflower. In China, I suppose everyone knows about this plant, for it has been cultivated and domesticated in China for thousands of years, and it has been recognized as a national flowers of China for nearly 100 years. But for the Westerners and for many other countries, very few people knows about this species. Even in Chinese English dictionary, it called Mayflower as a plum blossom or Japanese apricot. Actually, Mayflower is neither plum nor the apricot. So that is why I'd like to take this opportunity to make a presentation at this international symposium. I'd like more people to know about this species. The botanical name of the Mayflower is Prunus mume, or Armeniaca mume. It depends on different botanical classification system. It has been a valuable fruit and ornamental plants cultivated for more than 3,000 years in China. The top left picture shows that the stem of May were disintegrated in the Neolithic period or Neolithic age, which is about 7,000 years from today in Wujiang County, eastern China's Jiangsu province. This is also world's earliest discovery of the May stem. The upper right picture shows that the stem of May about 3,200 years ago were disintegrated in Yinshu Tomb in central part of the China's Henan province. The bottom picture shows that the May stem in Western Han Dynasty were disintegrated in Mawangdui, Hunan province for more than 2,000 years. All of this May stem disintegrated suggested that the ancient Chinese people began to eat May fruits with a long history and preserved them as an important fruit. Compared with other ornamental plants, the Mayflower has some very important features. The first, Mayflower is able to bloom in the snow, forming a very beautiful landscape. In terms of climate in different areas, it can flowering from December to early January in Guangdong, Guangxi, and Yunnan province. In February, along the Yangtze River Valley area. So these characteristics is also an important symbol of the formation of Chinese flower culture. The second feature of the Mayflower is it can release very pleasant aroma, fill the air with fragrance. According to our study for many years, the aroma components of the Mayflower are very good for people's health. In addition, the Mayflower has a wide range of uses. It can be used as a bonsai, or in Chinese, penjing, and it can build May Garden or May Valley and so on. It attracts tens of millions of tourists and travelers to the Mayflower Gardens each year in China. Mayflower can also produce as a cut flowers. It can make dried flowers which can use as a medicine and tea, perfume, air freshener. The fruit of May has many uses, such as fruit wine, sauce, candy, preserved fruits. It forms a large production industry. Now there are about 120,000 hectares in China. Now let me talk about why we conducted breeding for cold resistance. Mayflower is native to China. According to our survey for more than 20 years, there are 15 provinces and autonomous regions which are existing wild Mayflower. May is naturally distributed in Yangtze River Valley and south of China. As I mentioned, Chinese people loved Mayflower for thousands of years. Admiring and enjoying the Mayflower blooming in the snow has been a dream of the people in the north part of China. So introduce the Mayflower to growing in North China, especially in capital Beijing, has been carried out for hundreds of years. But unfortunately, the introduction is not successful. Therefore, cold-resistant breeding is one of the important goals for our flower researchers. This is a breeding strategy for cold resistance of flower. First of all, we collected the resources of more than 200 May cultivar, and it is related species such as mountain peach. The Latin name is Prunus davidiana, apricot, the Prunus armeniaca, Siberia apricot, the Prunus siberica, and so on. Then we use several methods to evaluate their cold resistance. In the course of studies, we selected 3 cold-resistant species as hybrid parents that are Prunus siberica, Prunus davidiana, and Prunus armeniaca. So we use this Mayflower to cross with those species. We set up crossing combination. After we got hybrid seedlings, then we conducted tests both in the laboratory and fields. We use morphological and molecular markers to identify authenticity of the hybrids. Then we conducted back crossing to get what we want, the ideal cultivars. Here are some of resources of Mayflower cultivars we have collected. We collected a total of 275 cultivars and set of a garden in our forest farm and selected crossing parents. We analyzed and evaluate the pistil and stamen fertility and cold resistance of Mayflower, as well as 10 related species. Here shows that we used May cultivar Danfeng Hou as a female and true May series cultivars as a male to conduct hybridization. So this is a cross compilation and seed seedlings rate of 10. From 2010 to 2017, we carry out 62 cross combinations and obtained more than 3,000 hybrid seedlings. Here shows our research team make a pollination in Qingdao, Shandong province. Because of incompatibility between embryo and endosperm in process of hybridization of Prunus mumma with Prunus sibirica and Prunus armeniaca, the phenomenon of early fruit drop is very serious. So it is necessary to rescue early embryo. Here shows that embryo rescue experiment 65 days after pollination. After years of hard work, we got new cultivar, Jiangrui Bai. So this is our first interspecific hybrid cultivar we made, which can tolerate the -25 to -26 degrees centigrade and overwintering in Beijing. These are some of other cold-resistant new cultivar that we have made. The top left picture shows the new cultivar which was crossed with Prunus cerasifera. The upper right picture shows that the new cultivar were obtained from hybridization between Prunus mumma and Prunus sibirica, as well as armeniaca. The top middle picture shows that the new cultivar, which was a hybridization between Prunus mumma and Prunus davidiana. Prunus davidiana is also called mountain peach. It is native to China. It distributed in the north and the northeast area. The new cultivar can be a survivor at the -30 degrees centigrade in winter. So we use this new cultivar to make a garden in Beijing. Here shows the May garden in Beijing Botanical Garden. It attracted hundreds of thousands of people to visit there every spring. There are 3 new hybrid cultivars planted in Jilin province, which are located in northeast part of China. The new cultivar can survive at the -38 degrees centigrade in winter without any freezing damage. So this is the first time in history that the Mayflower has been planted in the northeast province. This picture shows that Mayflower was blooming in the snow at the main city World Park, capital Beijing. Now there are 44 May gardens set up in Beijing. The Mayflower gardens have become beautiful landscape in early spring in Beijing. Here shows the map. Here the map shows that the new cultivar of Mayflower has moved northward for more than 1,500 kilometers. The middle line in the map is a northern boundary of the natural distribution of Mayflower. So this is a line. This is a northern limited of natural distribution of Mayflower. Now the new cultivars of Mayflower can be planted in northeast and northwest China. So here it should be said that we have made some achievement in breeding for cold resistance of Mayflower. But now the problem comes. All the hybrids from interspecific hybridization lost their typical May aroma. Hybrids between Mayflower and other Prunus species, such as Prunus armeniaca, Prunus sibirica, Prunus davidiana, Prunus cerasifera, and Prunus persica. So lost Mayflower fragrance means that the new cultivar will greatly reduce its ornamental value. Why do the hybrids between the species lost their fragrance? what are the typical aroma components of the Mayflower. So this has become the major issue of the cold-resistant breeding of the Mayflower. So this is the original intention of our research. So in order to determine the dominant fluorescent components in Mayflower, we conducted the quantitative analysis of main scent components by using GCMS for both emitted volatiles and endogenous substances of May cultivars, and it is closely related to 8 species such as almond, purple leaf plum, plum, cherry blossom, flowering plum, a mountain peach, Siberia apricot. So the main results as follows. The first, as a picture show, there are about 40 volatile substances belong to terpene, fatty acid, and benzoyl in Mayflower, which is the most one compared to other related species. The second, as the picture show, we detected benzoyl acetate account for about 90% in the relative content of the May volatile substances, but there is no benzoyl acetate in other related species. Then we analyzed the main scent component for emitted volatiles and the endogenous substances of the May flower, and it is closely related to 8 species. As the table shown, we identified that there are about 88 microgram per gram in endogenous substances of Mayflower, but they rarely exist in other related species. Meanwhile, we detected that there are about 4,850 nanogram per gram per hour in emitted volatile substances of Mayflower, but they rarely exist in other related species. So we determined the benzoyl acetate is a dominant component in main flora scent of the Mayflower. But how is the benzoyl acetate formed? In metabolism pathway of the flora scent, in study of Petunia and Calathea breviarii, we determined that the direct biosynthesis metabolism pathway of benzoyl acetate, as the picture shows, the benzoyl alcohol acetyltransferase catalyzes the benzoyl alcohol into the benzoyl acetate. So we identified the BEAT genes play an important role in the biosynthesis of flora scent in Mayflower. In order to explore how the BEAT genes regulate the biosynthesis of the benzoyl acetate, we suggested that the research should be conducted from the whole genome. So the May from Tibet was sequenced by the next generation sequencing. About 84.6% of its genome was assembled. We annotated 31,319 protein coding genes by RNA-seq. As the picture shows, we anchored the main genomic sequencing to 8 linkage groups constructed by applying RAD marker strategy. Then we constructed the main consensus genetic map. And at last, we published this study in Nature Communication. In May genome, as the table shows, we identified 34 beta gene family which expanded notably in May genome compared with other plants there are 16 bit gene family in apple genome, 17 bit gene in poplar, 3 bit gene in Arabidopsis, 4 bit gene in grape, and 14 bit gene in strawberry. So there's no bit gene family in rice. Meanwhile, as the picture shows, we also detected 26 of 34 main bit genes lay in cluster, the largest of which contained 12 members that were arranged in tandem in chromosome 3. Then we conducted the comparison of bit gene family in the genome of Mayflower and peach blossom. As the picture shows, we identified 32 May bit genes were arranged in tandem in the first branch of this phylogenetic tree, but only 6 bit genes were arranged in the peach genome. So in the summary, the expansion of the beta gene family might increase the content of the benzoyl acetate and be related to the special fragrance of the Mayflower. In order to verify this beta gene function in May genome, we cloned 3 beta genes from Sanren. This Sanren is a cultivar of Mayflower with white flowers. So we used an RT-PCR method. Then this gene will transfer it into the tobacco and the Arabidopsis for function verification. As the picture and the table show, the difference between the wild type and the transgenic plant testified that the beta gene could effectively catalyze benzoyl alcohol to benzoyl acetate. The highest catalytic efficiency can reach to 5.75 times than the control. So in conclusion, the study supposed that the expansion of tandem repeated of the beta gene family might catalyze the benzoyl alcohol into the benzoyl acetate and increase the contents of benzoyl acetate. This may be related to the special fragrance of Mayflower. So at the same time, we studied the expression of beta gene in different organs and tissues of the Mayflower. We cloned 3 beta genes, the beta 34, beta 36, and beta 37, and found it highly expressed in flowers, and their highest level were detected during the blooming stage. Overexpression of the beta 36 or beta 37 genes increased the benzoyl acetate production in the petal protoplast of the Mayflower, and the interference in the expression of those genes slightly decreased the benzoyl acetate content. However, the beta genes in peach blossom were mainly expressed in leaves and stems and could not be detected in flowers. So it is suggested that the expansion of the beta genes in the genome induced the characteristic components, the benzoyl acetate production in the Mayflower. Our study also observed the eugenol is another component of characteristic fluorescent in the Mayflower. We cloned 2 CF80 genes from the cultivar Sanren named CF81 and CF82. CF81 was highly expressed in the petals and the stamens, and this expression increased from the budding stage to the full blooming stage and decreased in withering stage. So it is constant with the patterns of eugenol synthesis and the emission. CF82 has a different type of expression pattern. Overexpression CF81 in tobacco result in eugenol biosynthesis in leaves. So it is suggested that the CF81 but not CF82 contributed to the eugenol synthesis in Mayflower. In order to elucidate the flora fragrance diversity of the Mayflower, the flora volatile emitted from 8 cultivars and 8 cultivars of May with white, pink, and red flowers were collected and analyzed using head space solid phase micro extraction combined with GCMS. 31 volatile compounds were identified, in which 10 phenylpropyloides were considered as the major components for the flora scent. Besides benzoyl acetate and eugenol, synamyl alcohol and synamyl acetate are also the characteristic components of the flora scent in Mayflower. Benzoyl acetate and eugenol were the characteristic components for white flower cultivars. Synamyl alcohol and synamyl acetate can only be detected in pink flower cultivars. The eugenol was the main characteristic component for red flower cultivars. Principal components analysis showed that the amount of benzoyl alcohol, synamyl alcohol, benzoyl acetate, eugenol, synamyl acetate, and benzoate could make these cultivars distinguishable from each other. The hierarchical cluster analysis of the flora scent indicated that the cultivars with similar flower colors were grouped together. So from synthesis pathway of the Mayflower, we can see the anthocyanin and the fluorescent related phenylpropyloides, that is eugenol and methyl eugenol in Mayflower, have the same precursor in synthesis pathway. As shown in picture, the amount of eugenol and methyl eugenol in pink and red flower cultivars were mostly higher than those in white one, while the amount of the benzoyl acetate in white flower cultivars were higher than those in other cultivars. The beta and CFAT, the enzyme activities of which have been verified in our work, both belong to the BHAD acetyltransferase family. As shown in synthesis pathway, acetyltransferase play an important role in characteristic flora synthesis in Mayflower. The above is a result of our research on flower fragrance through genomics. So this is our research team. That's all. Thank you for your attention.
Enormous work you have done so far with in-depth research on May, Prunus mume. I think it's very interesting result you have. I think we can have from now on to get some questions from floors or online.
Mr. Chairman, Professor Kim.
Yeah.
Professor Kim. Mr. Chairman.
Yeah.
Mr. Chen.
Yeah,
Mr. Chen, can you hear me?
yeah, I hear you.
I'm sorry. I I can't discuss with the participants because there is another a meeting at the last minutes I have to attend. So, if anyone is interested in my report, so please contact me.
Yeah.
Some of this my report have published a paper, and we also published a book, the genomics of Mayflower, but it is a Chinese version. We will translate into the English version.
Okay. Yeah.
So today I'm
Yeah.
very
Yeah.
sorry.
Yeah.
I
No
have
problem. Yeah, I have
no time.
already. Yeah, I have already got some information that you are very busy from this moment because you have another meeting at the university. I think, as you already mentioned and suggest that if someone has questions, please email him. Uh, Maybe we can support some email address on the website as well. So thank you very much, Professor Zhang, once again for your valuable and rich result. Congratulations. Let's give him a big hand. Yeah. Thank you very much.
Oh, thank you, Professor.
Yeah. Okay. The presentation has been scheduled for 1 hour, but as Professor Zhang mentioned just before, that he has a very important meeting, so that he has to leave this symposium. I think we proceed the next invited presentation, and then we will have a coffee break. All right. So maybe we have some more coffee break time because of Professor Zhang has finished a little bit earlier than scheduled. So we invite Professor Dr. Kim Sang-yong next presentation. His title is The Project Commercialization of Native Wildflowers in Korean National Arboretum. This presentation is originally is scheduled to give online, but now it is given from recorded version. I hope all the audience will understand it. So, is it ready now? Yeah, please go.
Hello everyone, I'm Sang-yong Kim, the Director of the Division of Plant Resources at the National Arboretum of Korea. I have been looking forward to meeting researchers from all over the world at this conference. It is too bad we can only meet online because of coronavirus. Still, I am happy to show this presentation about one of our projects, the commercialization of native wildflowers. These are some of the native flowers we are working to develop as new ornamental plants. Today, I am going to present what we are working on to make these beautiful wildflowers more accessible to the general public, and also to introduce our national beauty nationwide and worldwide. Currently, there are about 4,180 native plant species in Korea. However, considering that there are newly found species or unrecorded species every year, the actual number far exceeds the reported number. Meanwhile, some of these native plants are specified as rare plants by Korea National Arboretum. because of their population or habitat reduction. There are 571 taxa that are currently registered as rare species. One of our responsibilities as a national arboretum is developing techniques for conservation and propagation of these endangered species. There are also a plant group categorized as endemic plants. Their distribution is limited only to Korean Peninsula, and thus their propagation, utilization, and conservation is highly crucial. There are 306 taxa of endemic species across 7 genera. With the rising interest in ornamental plants, as we can see from increasing number of flower or garden shows in Korea, public interest for native plants is also increasing. According to our survey, native species accounted for 9.8% of the plants sold at flower market. In Korea, native plant sales have increased steadily over the last 5 years, from 2014 to 2018. As you can see from these graphs showing wildflower production, wildflower production quantity increased by 16% of every year, and the sales figure increased by 30% over the 5 years. Although gaining in popularity, native plants are still underused because of a relatively small market, nurseries with limited production area and capital, lack of information and familiarity with native plants, and the misconception that native plants don't have as much ornamental value as the non-natives. Thus, the use of native plants is less than 10% of all garden and landscape plants. There are about 593 species with ornamental value in Korea. For plants that have been produced more than 100,000 pots per year, are only about 98 species. This shows that there are still many more native species with commercialization potential. Therefore, based on this market trend and to promote the use of native wildflowers as garden or potted plants and cut flowers, Korea National Arboretum is currently leading the project Native Wildflowers to encourage the commercialization of wildflowers that are native to Korea. The project consists of 4 major steps. First, development of cultivation technology. Second, breeding of new ornamental plants. Third, developing model gardens. Fourth, Korean plant name index. Now, I'll go into more details of the process of this project. As mentioned earlier, the first step is the selection of new plant materials. They were selected with the help of an advisory group. And then the plants were identified and collected from their natural habitat. And the field and greenhouse trial were performed to decide whether the species is suitable for commercial production. Then the decision was made to either include or exclude the species for further development. An advisory panel for plant selection was organized, including professors, researchers, growers, and marketing specialists, such as landscape architects, florists, and flowering plant joint market. We are working with 46 advisors across 9 areas to discuss several issues on new ornamental plants, farmer selection, sales price of new ornamental plants, and many more. Evaluation criteria for the selection of target species are their ornamental value, usage, cultivation period, propagation efficiency, flowering control, cold or drought resistance, etc. At first, 87 species of native plants were nominated, and 30 species were finally selected after full review, including 24 herbaceous and 6 woody species. Here are some examples of our target species: Euphorbia jolkini, Corydalis speciosa, and Patrinia rupestris are native plants with beautiful yellow flowers. This picture shows Euphorbia jolkini in its natural habitat in Jeju Island. It has a high ornamental value and flowers in May in its natural habitat. In order to develop these target species as new ornamental plants, studies were done with their population distribution and growth environment. This shows a case study with Euphorbia jolkini. Distribution map was created and the number of plants and the GPS location was recorded for each population habitat. A trial plot was made at our center, and the field performance evaluation was carried out for the targeted species. The plant growth was monitored throughout the year and was compared with their growth in the natural habitat. Only the plants that retained their ornamental value in the field trial, according to their growth and flowering pattern in the trial plot, were selected for further study. For instance, Minuacia lericina flowered in May, but flowered once more in August at our trial plot. Phlox subulata is a species with similar growth pattern, but it flowers only in April. Minuacia lericina flowers only once in July in its natural habitat, showing that the flowering pattern can be significantly different under field conditions. Corydalis speciosa is another Korean native plant in the family Pumaraceae that has beautiful yellow flowers. It also grows naturally in Jeju Island. It has a long flowering period of 2 to 3 months, flowering from April to June, which is a big strength as an ornamental plant. Some of our target species have white flowers. These are Minuacia lericina, Arineria juncea, Leishmachia mauritiana, Aranchus etashipolius, and Exocoda ceratifolia. This is a picture of Minuacia lericina in the family Caryophyllaceae. It has a high propagation efficiency both from seeds or cuttings. It flowers from July to October in its natural habitat and can be a good alternative for garden plants such as Phlox subulata or sage. This is Arineria juncea in the family Caryophyllaceae. It has a high ornamental value because of its unique combination of grass-type leaves with white flowers that look like gypsophila, a popular ornamental flower. It also has a relatively long flowering period of 2 months from July to August. We have also selected 9 species of Veronica, which is a summer flower. According to Hawke, Veronica have a long flowering period and are easily cultivated, so they are often used as landscaping or cutting materials. Among Korean native Veronica species, Veronica kiusiana variety diamantiaca and Veronica busanensis are Korean endemic species. They both flower from July to August. Veronica kiusiana variety diamantiaca is a dwarf type, which is an advantage as an ornamental plant, especially when using it for potted plant. Veronica sinensis has a unique crawling growth habit and has a high environment tolerance. 3 species of Eschscholzia, including Eschscholzia angustifolia, Eschscholzia pensanensis, and Eschscholzia minima, are autumn flowers. Eschscholzia angustifolia and Eschscholzia pensanensis have a very high propagation efficiency. Eschscholzia angustifolia is registered as Korean rare species, and its leaves have a unique smell. Eschscholzia pensanensis is registered as Korean endemic species, which grows only in Byeonsan, Korea. The next step of our project is developing environmental and cultural control technique of the target species. Developing propagation technique is the first and foremost step. For propagating from seeds, the germination rate was observed for the selected species. For commercialization, germination rate of at least 70% is required. For those with low germination rate, the seed dormancy mechanism was investigated. For example, germination of Euphorbia jolkini seeds was observed in 2 different temperatures, at 15 and 25 degrees Celsius, and with gibberellic acid treatment. You can see that the germination rate dramatically increased on the 15 degrees Celsius and with gibberellin treatment. Propagation from cuttings is another crucial part, particularly for plants like Patrinia rupestris or Euphorbia jolkini, which generally takes 3 to 4 years to flower. When they are propagated from cuttings, they flower within a year. For these species, rooting and growth response was observed for different cutting collection time and rooting hormone treatment. Plug seedling production for potted plants is also a highly important part of native plant commercialization. There are several factors that can be manipulated in the greenhouse or nursery to control plant growth. We have investigated the effect of container size, soil composition, and seed number per cell on seedling growth for improved production efficiency. After the seedling stage, cultivation focuses on improving the ornamental value of the containerized plants. The pot size determines the ornamental values and morphological changes, and control plant growth. This example shows the effect of pot size on the growth of Eschscholzia angustifolia. For environmental and cultural control during the production stage, the effect of light, fertilization, plant growth regulators, and temperature is studied. This shows fertilization experiment for Euphorbia jolkini and Exocoda ceratifolia. The far left plant in each figure shows the control, and you can clearly see the effect of fertilization. Traditionally, native plant growers have focused on the garden plant production, but these days potted plant production is increasing. Therefore, the use of plant growth regulators such as paclobutazole and dominozide have become crucial for producing more compact plants, and planting studies using BAP are also in progress. Light condition is another important factor for plant growth. We are performing shade resistance experiment for the target species, and the results vary for different species. Most of our targeted species showed better growth under 0 or 30% shading. But in Ironcus etosifolias, plant growth was maximized under 60% shading. And the sun-exposed plants showed a compact growth characteristic, as you can see in this figure. These results are helpful when planting these species outdoors or for greenhouse growers. For some species, sodium chloride and cold tolerance have also been tested. Research on flowering physiology is essential in order to make year-round cultivation possible. Flowering is primarily regulated by vernalization or chilling treatment, photo period, temperature, and light. Flowering requirements can be categorized by combinations of the photoperiodic response and the vernalization effect. For example, our research confirmed that Patrinia rupestris is an obligate long day plant and also has an obligate cold requirement. The figure shows that it flowers only under long day conditions. Based on these results on propagation and cultivation, we are working on a plant cultivating guidebook. For our targeted species, as you can see from this example on Ranunculus crocylobus, this information can work as a basis for spreading our technology to native plant growers and consequently introducing and supplying the targeted species to the public. At the same time, we are also working on breeding new ornamental native plants, mainly to improve limitations of native species. The limitations of native plants include tall plant height, small flowers, short flowering period, limited flower color, and cultivation period. We are performing breeding studies to overcome these traits, and the current project focuses on Veronica species. Foreign cultivars of Veronica in the Yangtze flower market have increased significantly over the last 5 years, and now they are easily accessible to the general public. There are 25 species of native Veronica in Korea, and 9 species have been collected for study in this project. The first method of breeding is selecting from the natural habitat. By mutation fixation, through seed or vegetative propagation, our goal is to register new plant varieties. Examples of selective breeding include Veronica busanensis, for which we found a variation in flower color from purple to pink, and in plant form from crawling to upright. The second method is crossbreeding. Among 6 native species and 40 foreign species of Veronica, we have tested 94 combinations and selected 18 lines. In 2018, we have successfully crossed the native Veronica pyrethylina with the foreign Veronica spicata from a Nana blood pitch and named it Splenica. It has a hanging down plant form, oval leaf shape, light purple flowers, and uniform flowering in June. It is currently in the process for variety registration. Another method is mutation breeding using radiation. Native Veronica longifolia showed decreased plant length, increased leaf size, or different flower color after gamma ray irradiation treatment. We are planning to complete the variety registration process by next year and to supply these materials to the growers. This is a case study on hosta breeding from 2016. We have obtained various hosta variants from our research, and especially this patterned variant derived from hosta ingeri is one of the successful outcomes. The final step is putting together the cultural information for each selected species and supplying these species to growers and ultimately to customers. In order to observe the actual plant performance in the farmhouse setting and to confirm the productivity and profitability of the target species, farmhouse cultivation have been carried out since 2019. There are 5 farmhouses in 2019 and 5 were added in 2020, making it 10 farmhouses across the country. Because each farmhouse is in a different region and has a different facility and cultivation environment, it was recommended that farm owners select and cultivate 2 or 3 species out of 6 targeted species. This table shows that 3 groves in Pyeongchang, Yongin, and Gwangju have been cultivating and producing Minuacia lericina and Veronica busanensis. We have provided plant materials and technical training to the growers, and based on this, Minuacia lericina have been produced both as ground covers and containerized plants, and over 2,000 plants have been sold as a trial last fall. Based on these results, sales of Minuacia lericina have started in full scale this year, through online and offline stores as potted plants, and they are quickly going viral. They have been used in landscape planting for apartments as well. Minuacia lericina is a successful case of commercialization of a new native ornamental plant. The promotion of new ornamental plants and the sharing of research results have become very important, because there must be a demand for this information along with the actual supply of these plants. Therefore, we are working on making a leaflet and a guidebook of cultural manual for 20 selected species and planning to publish the first part by November this year. To share our research outcome with the public, we have been holding a wildflower exhibition every year. In January 2018, it was held in Daejeon, under the theme of wildflowers to meet the season. January is the season of the year when there is not much flowers around because of a freezing temperature in Korea. We picked January to demonstrate our achievement with controlling the flowering time of native flowers. In 2019, it was held in Seoul at Seoul Station, where there is a large floating population of about 1 million people per day, so that we can effectively show our results and share the opportunity to experience wildflowers with more people. This year, it was held outside because of the COVID-19 at Korea National Arboretum. 47 species, including 12 targeted species, were planted, and this exhibition is also available online through YouTube. Our project is not just limited to the research on individual plants. We are also working on ways to actually utilize these plants. Therefore, this project includes gardening study, where we select gardening plant materials and develop model gardens and apply them to an actual garden setting. We work on planting the selected native plant materials to various types of gardens, such as rain garden, wetland garden, roof garden, or rock garden, and study how they perform in a garden setting. This shows an example of our model garden design and planting scheme. Lastly, we are also working on the Korean Plant Names Index, KPNI. This is a database of the names and associated basic bibliographical details of all Korean vascular plants. Its goal is to eliminate the need for repeated reference to primary sources for basic bibliographic information about plant names. These are some of the booklets we have published as the outcome of our work on Korean Plant Names Index. Thank you so much for your attention. If you have any questions, I will be happy to answer them.
Thank you very much. Dr. Kim, I forgot just introducing the presenter, Dr. Kim. I will introduce him just now. He is a senior researcher of Korea National Laboratory, also leader of Division of Plant Resources. The task of this Plant Resources Division are mainly development of new ornamental plant and also development of Korean native plant database and wild useful garden plant collection and mass propagation and conservation of rare endemic species, Korean plant names index, and other research of plant for climate changes, as he just mentioned and presented at the end of his slide presentation. Thank you very much once again to Dr. Kim. Let's give him a big hand. Although he's not able to receive on some questions, both Professor Zhang as a plenary speaker, a keynote presentation, and also invited speaker, just Kim has not able to receive any questions from on board or on the floor or online. So I'm sorry to give such a news. From now, I think we have enough time to have a coffee break. Actually, this is scheduled till 4 o'clock in the afternoon. Now it's 3:35, so we will have a coffee break until 4:15 p.m. Eastern hour. So let's have a break time. Thank you very much. We will continue later.
So many doors in this place. Where are they all connected to? For whom was the first step for? Newness becomes another culture. Trace becomes another story. The breath of this city that has ran without resting will tell you. Easy to reach, being closest to the Skyway. In this city full of life, the fanciness that blends with the old will satisfy anything. The magical scenery you don't know how you'll meet will come to you as a heart-touching moment. When you stay at a place for you prepared here and there, you will then know why they call this place Always Incheon. See, right now, right here, right this moment, the city breathing alive. To wherever you want, over time and space. The first in Incheon will be a specialness over the newness. Always Open City Incheon. So many doors in this place. Where are they all connected to? For whom was the first step for? Newness becomes another culture. Trace becomes another story. The breath of this city that has ran without resting will tell you. Easy to reach, being closest to the Skyway. In this city full of life, the fanciness that blends with the old will satisfy anything. The magical scenery you don't know how you'll meet will come to you as a heart-touching moment. When you stay at a place for you prepared here and there, you will then know why they call this place Always Incheon. See, right now, right here, right this moment, the city breathing alive. To wherever you want, over time and space. The first in Incheon will be a specialness over the newness. Always Open City Incheon. So many doors in this place. Where are they all connected to? For whom was the first step for? Newness becomes another culture. Trace becomes another story. The breath of this city that has ran without resting will tell you. Easy to reach, being closest to the Skyway. In this city full of life, the fanciness that blends with the old will satisfy anything.
Okay. The coffee break is over, so we continue the oral presentation. We only have 3 oral presentations for this symposium today. After 3 oral presentations, the closing ceremony will be scheduled. So please keep the seat and also keep online for the finish of this symposium. Okay, next speaker will be Wen Sing and their group from College of Landscape Architecture, Central South University of Forest and Technology, Henan, China. The talk will be genome-wide analysis of HSF family transcription factors and their responses to heat stress in Rosa sinensis. So, please, Wen Sing, are you ready?
I'm ready.
Okay, please start.
Yes. Good afternoon, everyone. My name is Xing Wen. I'm from Central South University of Forestry and Technology in Changsha, China. I'm pretty glad to give you a presentation about my research on genome analysis of HS transcription factor and their response to heat stress in Rosa sinensis. Rose is one of the most important ornamental crops in the floriculture industry. The optimum temperature for growth and flowering of rose was from 22 to 26 centigrade. High temperature restricts the growth and development of rose. Previous studies proved that transcription factors induced by heat stress are the crucial factors in the transcriptional regulatory network of plants. Among these transcription factors, HSFs act as the core regulator of the heat stress response. They mediated the rapid accumulation of many important function proteins induced by heat stress. So, identification of the key HSF genes in heat stress response is important for promoting heat stress tolerance in rose. However, the information about the HSF gene family in rose is currently unavailable. Recently, the whole genome sequencing of Rosa sinensis Old Blush was completed and published. It provides us opportunities to comprehensively investigate the HSF gene family in rose. In our study, we carried out the genome analysis of HSF genes in Rosa sinensis Old Blush. The detailed information and phylogenetic analysis of these HSF genes were conducted. Furthermore, combined with comparative transcription analysis, the heat stress responsive pattern of HSF genes were elucidated, and the important candidate HSF genes in transcriptional regulatory network of heat stress response in rose was identified. Our study will lay the foundation for genetic improvement of heat stress tolerance in rose. In our study, 19 non-redundant candidate HSF genes were identified in the genome of Rosa sinensis Old Blush. According to their location order on the chromosomes, we named these genes from RCHSF1 to RCHSF18. The classification and chromosome location, as well as the physical and chemical properties of all these 18 HSF genes were analyzed. These genes were distributed on all 7 chromosomes of Rosa sinensis, but the numbers differed on different chromosomes. The chromosome 2 contain the largest number of HSF. The lowest number was found in chromosome 1. The physical and chemical properties of all 18 HSF genes were shown in this table. The CDS length of all RC-HSF genes ranged from 618 BP to 1,578 BP. The protein size ranged from 205 amino acid to 535 amino acid. The predicted isoelectric point varied from 4.71 to 8.71. The molecular weight ranged from 23.83 kd to 58.48 kd. In order to analyze the phylogenetic relationship and classification of HSF genes in rows, 21 AT-HSF protein from Arabidopsis and 25 OS-HSF protein from rice were used to construct the phylogenetic tree together with 18 HSF protein from rows. The result shows that all 18 HSF protein were classified into 3 main groups, Group A, B, and C. HSF-A group was the largest group and contained 11 members. HSF-C group was the smallest group, only has 1 member. Furthermore, the 3 main groups were divided into 15 subgroups based on the phylogenetic analysis with Arabidopsis and rice. The largest group A was classified into 8 subgroups. Subgroup A1, A4, A6 has 2 members respectively. Subgroup A2, A3, A5, A8, A9 has 1 member respectively. The group B was further classified into 4 subgroups. Subgroup B1, B3, and B4 has 1 members respectively. Subgroup B2 has 3 members. The gene structure and the conserved motifs were also analyzed in our study. The intron and extron structure of 18 HSF genes in rows were shown in this picture. We found 5 genes with 3 exons, and the other ones all had 2 exons. In addition, we tried to identify the conserved motifs in complete amino sequence of 18 HSF proteins. 10 conserved motifs were selected and analyzed. Most HSF gene proteins had motif 1, 2, and 4. They were located at the N-terminal, and they represented the HSF domain and the DBD domain. Motif 3 and motif 5 was found to be connected to motif 4 by a flexible linker of 15 to 80 amino acid residues. They are the HR, A, and B region. We also found 6 proteins with only motif 5. These 6 proteins belonged to group B. Motif 9 represented the nuclear location signal domain. We found 10 proteins with this domain, including 1 member from group C, 1 member from group B, and the other 8 members are all belong to group A. Motif 7 and motif 10 were found to be located at the C terminal. They represented the AHA domain. We found 5 members from Group A with these two domains, including HSF12, HSF13, HSF18, HSF9, HSF5. Among these HSF protein, HSF5 and HSF12 has two motif 7 and motif 10. In addition, in order to reveal the expression pattern of HSF genes and heat stress, we compared the transcriptional level of HSF genes in treatment of half an hour, 2 hour, 6 hour, and 12 hour of heat stress treatment in Rosa sinensis Slater's Crimson China. The results showed that 12 HSF genes were regulated by heat stress. 11 of them were upregulated, and only 1 was downregulated. 6 upregulated genes were induced from half an hour of heat stress treatment. The other 5 HSF genes were upregulated from 2 hours of heat stress treatment. Furthermore, we construct the phylogenetic tree to elucidate the relationship with the 12 HSF genes in Slater's Crimson China regulated by heat stress and 18 HSF genes in Old Blush. The result showed that 12 HSF genes regulated by heat stress in Slater's Crimson China belonged to 2 main groups, including Group A and Group B. 5 members in Group B were further classified into subgroup B1 and B2. All of them were upregulated from half an hour of heat stress treatment. Among these 5 members, Contig 12,705 and Contig 60,744 and 44 were most significantly upregulated by heat stress. These 2 genes were much closer to 2 members belonged to B2 subgroup, RCHSF1 and RCHSF17. 7 members in Group A was further classified into 6 subgroups, including subgroup A2, A4, A5, A6, and A9. Contig 81,627 was much closer to 1 member belonging to A9 subgroup, RCHSF10. It was the only 1 gene downregulated by heat stress from 6 hours of heat stress treatment. Contig 113, 972, and Contig 245, 724 was much closer to 1 member belonging to subgroup A2. They were upregulated from half an hour of heat stress treatment. The other 4 members from subgroup A was all upregulated from 2 hours of heat stress treatment. Among these heat-regulated genes, the expression level of Contig 245, 724 was the highest one of all. So, RCHSF16, RCHSF1, and RCHSF17 may act as the crucial factors in heat stress response in rows. The function and mechanism of these 3 genes will be further studied. In summary, 18 HSF genes were identified in the genome of Rosa sinensis Old Blush in our study. They are assigned into A, B, C 3 main group and further divided into 15 subgroups. 12 HSF genes were found to be regulated by heat stress. 11 of them was upregulated. Only one was downregulated. The downregulated HSF genes was belonged to subgroup A9. 2 members from subgroup B2 and RCHSF1 and RCHSF17, as well as 1 members from subgroup 2, RCHSF16, were most significantly upregulated by heat stress. These 3 genes may act as the crucial regulators in heat stress response in rows. Our studies were supported by Natural Science Foundation of China and the Natural Science Foundation of Hunan Province. We also thank Wuhan Aijin Book Biotechnology Institute for technical support. That's all of my presentation. Thank you for your attention.
Let's give some questions. Any other questions? On genome-wide analysis of heat shock factors. No questions on the floor or online? Okay, let me ask you one question about your presentation. You mentioned 18 RCHSF genes found in Rosa sinensis, cultivar name Old Blush, right?
Yes.
Yeah. How many chromosomes of 18 RCHSF genes are located on how many chromosomes?
They located in 7 chromosomes in rows.
Among
7 chromosomes.
Among 12 or how many?
7.
Yeah. What's the chromosome number per genome of Rosa sinensis?
7 chromosomes. The Rosa sinensis has 7 chromosomes.
Okay. Okay.
And the HSF genes distributed in all 7 chromosomes.
Okay. Is it already full sequenced or not?
In our study, we investigate the CDS domain, the CDS of the HSF gene.
Yeah, you have analyzed this.
Not the whole, not the DNA sequence.
Whole genome sequencing. Eh? So
yes.
Do you know any information about the whole genome sequencing on this taxon, Rosa sinensis, in China?
No.
No. Okay.
Yes, published.
Yeah, I have heard that the BGI group, Beijing Genome Institute group, has been doing many plant taxa for whole genome sequencing so far. That's why I ask you about this Rosa sinensis, which is important in China because of the originated in China. So maybe BGI already did, or other groups did, some of whole genome sequencing of this species.
Sorry, I.
Okay. Okay. Are there any other questions? No? Okay. I think there's no questions. Thank you very much once again for nice presentation. Let's give a big hand. Thank you. Next presentation will be given by Wen Xing from Beijing Forest University. The same group as Qixiang Zhang's laboratory. The title of presentation is Dissecting the Genome-Wide Evolution of NUDX1 Gene in Geraniol Biosynthesis of Rosa sinensis. Are you ready? Okay, please start.
Good afternoon, everyone. My name is Yu Han. I'm from Beijing Forestry University. Thanks to the organizing committee for giving me this opportunity to share my recent research work. My topic is Dissecting the Genome-Wide Evolution of NUDX1 in Geraniol Biosynthesis of Rosa sinensis. Welcome to have academic exchanges with me via email. I focused on the formation mechanism of the important ornamental trees in rose. Rose is one of the most important ornamental plants because of its pleasant flower scent, continuously flowering trait, variable petal colors, and easy to breed. Today, I'd like to report to you that during the COVID-19 pandemic period, when I was unable to work in the laboratory a few months, I mainly used bioinformatics to do some research on the rose fragrance. Flower fragrance breeding is an important direction of rose. Until now, more than 400 kinds of volatile compounds have been identified. Most terpenes compounds, especially monoterpenes, are highly volatile and widely used in plant essential oil production, and because of its unique organoleptic properties and extensive medical effects, terpenes compounds have been highly applicable in foods, cosmetics, and clinical treatment. Geraniol is an acyclic monoterpene alcohol compound. It has a gentle, sweet smell of rose, which is one of the main ingredients of rose essential oil and mainly synthesized through the MEP pathway. One study found that the NUDX hydrolase RHNUDX1 is part of a pathway for the biosynthesis of free monoterpene alcohols that contribute to fragrance in rose. The RHNUDX1 protein shows geranyl diphosphate hydrolase activity in vitro and supports geranyl biosynthesis in planta. There is a special pathway for the geranyl synthesis that does not depend on TPS, terpene synthesis, in rose. Phylogenetic analysis at the genome scale among different species can reveal gene evolutionary history and functional divergence. It also can provide a deeper understanding of the genomic evolution among different species. The increasing availability of the high-quality genomic resources make it possible to comprehensive analysis the evolution and the function diversification of NUDX genes in plant genomes. For example, we used the phylogenomic Sentinel network of 35 plant genomes to clarify the relationship between R2, R3, MYB gene evolution and function, and predicted that RCMYB1148 from the Rosaceae specific cluster functions in monopropanoid pathway. So, we solved three questions. 1. why the NUDX1 from rows can participate the geranyl synthesis. 2. Can we use the phylogenetic analysis method to reveal the evolutionary history and the functional divergence of NUDX1? 3. Is there any other species geranyl synthesis related to NUDX1 instead of TPS? At the first, 115 species have been selected to construct a phylogenetic tree from 130 species, which genome data has been published. The 115 genome data have been used to analyze pairwise whole genome comparisons to detect all centenary blocks. A total of 3,794 NUDX and 92,260 ages have been detected in 115 species. The ages mean pairwise syntenic connections between genes. 48 centenary clusters were obtained with a K-click population clustering method implemented by SunFinder to further clarify the evolutionary events affect the NUDX homologous in various plant species. We developed clusters for monocots, roses, and asterisks. In this figure, we find that the lineage-specific clusters of monocots were the most. Let's talk about the NUDX gene family in Arabidopsis. There were 29 AT NUDX, and many substrates have been predicted as the homologous of RH NUDX1. AT NUDX1 belong to centenary cluster 38, and its substrate is 8-oxo-DGTP. Through analysis, the 48 clusters, we found 80 NUDX which have the same substrates may be assembled in one centenary network. These centenary net clusters can help us predict the functions of RC-NUDX. There were 42 RC-NUDX in Rosagenesis. RH-NUDX1 has 6 tandem duplication homologous in chromosome 2 and 3 tandem duplication homologous in transchromosome 4. Only this gene from chromosome 4 was in the cluster 38, means it has centenary relationship with AT NUDX1. The spatial temporal expression patterns of tandem duplication genes RC-NUDX1A to RC-NUDX1F consistent with the content changes of geranyl in Rosagenesis. These genes have no collinearities with other NUDX in 115 species. Through analysis, the phylogenetic relationships with all NUDX, we found NUDX from the same cluster contributed in the same big branch. In this branch, except the genes belong to cluster 38, we found a small branch, which contains RC-NUDX1s and 2 tandem duplication NUDX from Rosaceae Fragaria vesca and ribose oxidantase. RCNUDX1s from chromosome 2 were closer to 2-FV-NUDX than this gene. To sum up, there were 42 NUDX in Rosagenesis. The RCNUDX1s which participate the geranyl synthesis have no collinarities with other NUDX in 115 species. The unique geranyl synthesis pathway of Rosagenesis may be happened in Fragile vesca also, but it needs to be tested and verified. So we predicted the RCNUDX, which participate the geranyl synthesis, may be derived from protein mutations and transposon-mediated tandem duplication formation. About this study, we ongoing enzyme activity experiments for screening the key amino acids of NUDX gene, an enzyme for geranyl synthesis. At the end, thanks to the support and help from my postdoctoral mentor and leader, Professor Qixiang Zhang, student Jia Yaoyu, my collaborator Professor Tao Zhao, and my lovely leaders and colleagues. Thanks to the National Natural Science Foundation of China, the COVID-19 will eventually end, and the research needs to be continued. Looking forward to see you next time. Thank you.
Yeah. Thank you very much, Yuhan. It's very nice to have such a result and information to all of us who are mostly interested in ornamentals, but I don't think many of them are much interested in the genetics or molecular biology levels. Anyhow, but although they are not highly related to the research that you are now doing, but we can receive some questions, maybe some general questions also available on the floor. No? I think let me give you one general questions on Rosa sinensis about your result. You have mentioned that the RC NUDX1 genes are found in chromosome number 2 and chromosome number 4 that are mainly only located on 2 chromosomes. Yeah?
Yeah.
So then which number of genes or which NUDX1 genes is or are most strong expression activity in which chromosomes?
The NUDX genes, I think it's NUDX genes in chromosome 2.
Mm-hmm. So that you mean chromosome number 2 possess the most strong expression genes for NUDX1.
Yeah.
Right? Yeah. How many copies? I mean, what do you think about the genome evolution of Rosa sinensis? Are they duplicated or triplicated history? Or have you found any information about the evolution of Rosa sinensis?
The NUDX1, which we predicted in the geranyl biosynthesis, is in chromosome 2, and it has six tandem duplications.
I see. Okay. Yeah, I think you mentioned at the last PowerPoint that, but one of the participants asked you what is next step for your research based on this presentation result.
Please.
What is your next research purpose or steps? What do you want to do for next step?
Yeah, we ongoing the enzyme activity experiments. I will examine the amino acids activity of the RCNUDX1, which we predicted which can function in the germinal biosynthesis. We have obtained the protein, and we plan to use the mutation, STAT mutation, to detect this protein's function.
Much once again. Thank you.
Thank you.
Okay, I think the group of Beijing Forest University, the laboratory of Professor Xiang Zhang has been conducted a lot of work on molecular biology. I have seen many reports already in high standard international journal. Okay, last presentation will be given just from now. LED light improves post-harvest quality and longevity of cut rose flowers, cultivar Lovelty Lydia, from Department of Plant Biotechnology, Sejong University, Seoul, Korea. The presenter is Suang Tuyat Thai. This presentation is ready, just not online, but recorded version. Is it now ready? Okay, start.
Hello everyone, my name is Sung from Sejong University. Today I would like to talk about the topic, LED light improves post-harvest quality and longevity of cut lovely Lydia rosa hybrida rose. Contents of my presentations including introduction, material and method, results and conclusion. First of all, I would like to talk about the introduction. As we know, cup rose generally show a high deterioration rate and short phase life after harvest. Post-harvest techniques such as preservative solutions, antimicrobial agents, cold storage, and light irradiations have been applied to decrease deterioration rates of cup rose after harvest. But recently, chemical treatments for improving the phase life of cup rose is becoming less accepted due to other undersized residues and the environmental pollution. Whereas light irradiation methods are attentive because of residue-free and high efficiency in delaying germination symptoms and extend the shelf life of horticultural products such as fruits, vegetables, or cut flowers. However, there has not been a lot of research into the effects of simultaneous treatment of UV and LED light on post-harvest quality of cup rose flower. So in this study, we investigate the effects of UV and LED light treatment on raised life of cup rose to develop an effective and eco-friendly technology for improving post-harvest quality of cup rose during export process. Second, I would like to talk about material and method. In this study, we use cup rose Lovelty harvest from a commercial greenhouse in Changsu in July 2017. For the treatment, cup flower were treated with UV and blue LED light. Control flower were placed in same condition without any lighting system and then uncut flour in the box will save in 4 degrees and 50% of relative humidity in that condition for export simulations for 48 hours. After 48 hours, we measure internal and external quality of cut flour, some physical factors, water relations, and stomata characteristics. Our results indicate that treatment with UV and LED light delay senescence symptoms in cut flour, such as decreased yielding and abscess in beta compared with control flour, and significantly increase the baseline of cup rolls more than 2 days compared with control flour. Treatment with UV and LED light significantly increase flour opening and maintain chlorophyll content in leaf of cut flour compared with control flour. UV and LED light treatment also decrease bacterial population at cup roll stamen after 6 days of the treatment compared with control flour. The treatment also improves water uptake of cut flour during baseline compared with control groups. Treatment with UV and LED significantly maintain initial fresh weight and positive water balance of cut flours longer than control flours. In this study, we measure density stomata and stomatocyte in the leaf of cut flours. Our results indicate that under light condition and dark condition, the stomata opening rate in UV and LED treated flour are smaller than control flour, resulting decreased transpiration rate in leaf of cut flours. In conclusion, UV and LED light treatment delay senescence symptoms and extend the baseline of cup rolls by 2.9 days compared with control flour. UV and LED treatment also decrease bacterial contamination at cup roll stamens and then improve water relations of cup rolls during baseline. The treatment also decreased stomata opening rate and improves stomata function, resulting reduced water loss during baseline. Our results indicate that lighting system is an effective method to improve possible quality and longevity of cup rolls that are exported overseas. I have finished my presentation. Thank you very much for your listening.
Okay, thank you very much. Whole presentation, including keynote speaker and invited speaking, and also all the oral presentation has been finished for the ISGO 2020 symposium. Thank you very much for all participants online and here in also on the floor. Session 4 is officially finished, and we will continue the closing ceremony. Thank you very much for your cooperation.
Thank you, Professor Ki-byung Lim, to lead us for wonderful session 4. Good evening, everyone, for ISGO 2020. Thank you for joining the farewell ceremony of the symposium. I'm very glad to finish this symposium without any big accident, and it was very exciting experience, and I'm sometimes very nervous with internet or some sound matter. But I hope you all enjoyed this wonderful hybrid symposium. And I hope you all understand if we give you some inconvenience during the conference. So now I would like to announce the start of the farewell ceremony. First of all, I would like to briefly review the last 3 days of ISGO 2020. Starting with registration and welcome gathering, we had 4 sessions with great speakers and wonderful chairs. We also had a very special group photo time here at venue combined with Zoom participants. We had a wonderful gala dinner here at Korean traditional venue. And I'm very sorry that we could not all together in person at this moment. I hope we can share our memories with the photo later. We had total 112 people here at this venue during 3 days, and 106 people on Zoom from 6 different countries, including Korea, United States of America, China, Indonesia, India, and Portugal for this symposium on real time. And there are pretty more participants watching with YouTube and Darkest. Thank you for all participants. Okay, next, I'd like to proceed to ceremony for delivering appreciation to the convener, Dr. Byung-Yong Jung. ISHS medal will be awarded to the convener. Dr. Ki-byung Lim would provide the medal instead of the chair of ISHS. Please welcome the convener and the chair with a big hand. Okay. Now, Dr. Lim, he sent the ISHS medal to the Dr. Byung-Yong Jung. Congratulations. This is the medal from the ISHS. Okay, so we are now taking a photo. Congratulations again. Okay, and here is the flowers. Thank you. Okay. Usually this was offered from the ISHS chair in previous conference, but at this moment, because of COVID, it's very difficult. Okay, so now we will finish, yes. And we have Young Mind Awards. We had so many young participants along with young professionals. So ISHS provides Young Mind Awards to the best author and poster presenters who are a junior scientist and the presenter and first author of the submitted manuscript. So this Young Mind Awards winner will be recognized from ISHS website along with the Chronica Horticulturae, and it will be great accomplish for their scientific journey. So please turn on the Zoom also because we have international presentations winner. So the winner of the best oral presentation is Yu Han from China. So can you ready, Yu Han? Can you connect to the Zoom? Yes, here is Yu Han. Maybe you can remember her. Okay, now our convener, yes, send the medal and flowers to Yuhan, and we will definitely send. Yes, we will definitely send these medals to Yuhan with international email. So congratulations, Yuhan.
Thank you. Thank you. It's so surprise to me.
Thank you for your preparation and the nice presentation. Okay, and the winner of the best poster presentation is here, Aram Jo from Seoul Women's University, Korea. So, yes, here is Aram Jo here. Congratulations, and let's give them a big applause. Okay, now our convener, he gave her the ISH's medal and flowers. Okay. And we can have a photo time, so can Yuhan attend the photo? Okay. Very good. So congratulations again, Yuhan and Aram. Thank you. Okay. Okay, and now I would like to proceed to ceremony for delivering appreciation letter to sponsor. Actually, in the past conferences, we invited all sponsors to the venue, and we sent some medals directly. But due to the COVID-19, sponsors could not come here at the venue. So I conveyed the expression of the appreciation to the sponsors with this appreciation letter. So we are appreciated to Korean Society for Horticultural Science and Korean Society for Floricultural Science, Korean National Arboretum, and the Korean Federation of Science and Technology Society, Incheon Metropolitan City, and Gyeongsang National University, World Flower Company. So thank you very much to support our conference. Thank you. And now I would like to briefly talk about the result from the business meeting. This morning at 8 o'clock, we had a business meeting during the ongoing Third International Symposium on Germplasm of Ornamental. Professor Byung-Yong Jung was elected chair of the work group Ornamental Genetic Resources to a new term again. So, unfortunately, until now, no one has manifested interest to organize the next symposium in 2024. So we will try to find an organizer as soon as possible. So please give him a big applause for the next chair again. Okay, finally, now we will move on a short closing speech from our convener, Professor Byung-Yong Jung. Please give him a warm applause again.
good evening, afternoon, or morning. Dear respectable ISGO 2020 participants, we are here for the closing ceremony of the third International Symposium on Germplasm of Ornamentals held in Incheon, Korea. It was my great pleasure to conclude 2 days of first tried, not so familiar to none of us, a hybrid ISHS symposium without any big issues. Despite many hurdles we faced, even during this symposium, I personally evaluate that we had a quite successful symposium overall in terms of attendance, discussion participation, and execution of the program as planned. We had some unexpected technical pitfalls or irregularities, even when all our staff was on the highest alert throughout. These issues need to be considered and improved at the planning stage of future symposia. I would like to express my sincerest gratitude for all speakers, presenters, and the session chairs for their excellent talks, posters, and the session management. You guys did it very well beyond my expectation. I am so grateful for all the support from the ISHS, KSHS, KSFS, and the members of the organizing and the scientific committees. The financial support from the Korean Federation of Science and Technology Societies, Korean National Arboretum, Gyeongsang National University, Incheon Metropolitan City, and World of Flower Company made this symposium reality. Thank you very much all again. I thank for the dedication and the sacrifice by secretaries, editors, technical staff, and the students in making of the ISGO 2020 feasible in very difficult times. Dear participants, as all of us now know that we can continue our scientific activities using multicasting connectivity, let's try our best to keep our ISHS programs rolling. I really miss you. Stay safe and healthy always. Thank you very much.
Okay, so now our editor just instantly suggests me to take a photo together. So could you just come into the stage? In that case, maybe our participants through the Zoom, we can join. Okay. So through the YouTube, you can see the live situation here at the venue. So we will have some photo time together. So can you show the stage? Okay, perfect. We have beautiful flowers here on the stage. Our sponsors, KSHS and the Human Plant Environment Conference, they just to send us these beautiful flowers. Okay. So I also would like to join them for a photo shot. Okay. Okay, thank you so much for all the participants. You see here are lots of people just to keep stay at this venue until the end of our last closing session. So I'm really appreciated for your all cooperation and participants for ISGO 2020. Okay, so thank you again for all your outstanding contribution and cooperation. And I hope to see you again in 2024. Thank you.